and welcome to our Cardiac Safety Symposium on behalf of the team of Nanion Technologies. So we are happy to have you here today. And before we start, I would like to give you some general information about the symposium. So first, your video will be by default off and your audio is muted. The sessions of this symposium are being recorded and will be later available on demand. So by participating in this webinar, you consent that this session is being recorded. There will be a Q&A session available after each presentation. And we invite you also to write your questions in the chat during the presentations. So at the end of each presentation, you will have the possibility to raise the hand in the option that Zoom gives us. And you can also ask the question yourself if you would like to. Otherwise, the moderators will read them out loud. Before the break, we will have a poll. And at the end of the symposium, we will have a survey from which we would appreciate your feedback. Okay, so without further ado, so we can already start with the first presentation of the day. And it is my pleasure to introduce my colleague, Sonja Stölzle-Feix. She is the Director of Scientific Affairs at Nanion Technologies and also the Product Manager for the FLEXcyte 96 and the FLEXcyte. So here we have her.
Thank you, Yeimar. I'm also happy to welcome everyone to our second Cardiac Safety Symposium here. So this is the second of the annual series. We had our first symposium in 2021. So, well, over the years, we actually did. We did come a long way over the last couple of years. So we, and I'm speaking here on behalf of the global cardiac researchers and the cardiac safety community. We had quite a lot of recent publications from electrophysiologists looking at iPSC-derived cardiomyocytes and native cells, looking at patch length measurements using the SEPA protocol, best practice publications derived from the SEPA studies, and also calibration of proarrhythmia risk prediction models from the FDA. Also news from the implementation working group regarding the new ICH E14 S7B guidelines, just to name a few examples here. And we will also hear from some of the authors here today. So taking a step back, more than 10 years ago already, Yamanaka et al. Earned the Nobel Prize for the discovery that mature cells can be reprogrammed to become pluripotent. And this discovery changed a lot. Also, of course, for cardiac applications. So now I have a little interesting information collected here, which is the development of peer-reviewed original articles around stem cell-derived cardiomyocytes over the years. So obviously from 2012, there was a steep increase of the number of papers published per year. And most of the publications come from the US, but also quite a lot from China, Germany, and then Japan as well. Here you see the distribution across countries. So about 40% of the articles come from the US. Furthermore, more than 70 countries contribute to these publications, which means also more than 20,000 researchers contribute to all of these manuscripts. Now a shout out here to Artem, who is running the ionchannellibrary.com website. He was doing this research here on the literature, and you will find a lot more meaningful and interesting information on his website, so check it out. Now, quite some of those publications are authored by researchers who are also part of the HESI Cardiac Safety Committee. So the mission here of this committee is to improve public health by reducing cardiovascular related side effects from drugs, and to develop innovative approaches to support cardiac safety measurements. In the stem cell working group, which is co-chaired by Ksenia Blinova from the FDA and Godfrey Smith from Clyde Bio, University of Glasgow, there is currently the chronic cardiotox study ongoing, and we will hear about this study in multiple presentations today. So, speaking about innovative approaches and technologies, I would like to highlight this video protocol collection here from Trove. So the guest editors here were Ksenia again from the FDA, and Jennifer Pearson, who is an associate director at HESI. And this collection here addresses different cell models like single cells, 2D or engineered heart tissues, primary cells, iPSC-cardiomyocytes as well. And the approaches and assays reveal diverse endpoints, such as contractility, electrophysiologic readouts, or calcium fluorescence. Today we will hear a bit about FLEXcyte mature contraction force measurements as well, for example. So coming back to the HESI chronic cardiotox study, these are the compounds being investigated in the study, listed here with their mechanism of action. And we will hear from Khuram about the study efforts itself, but also we will hear from Bettina about the chronic toxicity and effects of those compounds on a cardiac cell model. And now, in order to pave the way for some of the results being shown today, I'm making a little detour, and I give you a brief introduction to the company I work with, Nanion Technologies, and also to the portfolio and technology that we develop and provide. So, well, we also came a long way. We celebrated our 20th anniversary last year, and starting in 2002, we developed automated patch lamp devices like the Porter patch, patch liner, or the Synchro patch, and also cell analytics systems like the CardioCyte and the FLEXcyte. And these systems are also part of the HESI study, for example. Now, as I said, we will hear about some contractility measurements today with CardioCyte, FLEXcyte. We will also hear about some synchro patch measurements, some automated patch clamping today. And now I'm really happy to announce a new development that Nanion is just around the corner to launch. This is the Atlas system. Now, the system is a impedance-based cell analysis system in a 6x96 well format. It allows for real-time cell analysis in a label-free manner. It automatically is graphing your results, and in general, it is a very easy-to-use system. So with the Atlas, you can tackle a huge variety of research areas, such as immuno-oncology, cell proliferation, or CAR T cells. So anything which is important for cancer research, for example. Also virology, GPCR, or barrier functionalities, just to name a few here. So regarding the setup, you have the recording unit standing in your own incubator. Then there's a control unit, a laptop coming with it, and a barcode scanner for your measurement plates. And the system actually is capable of deciphering complex cell responses in a continuous manner. So, for example, in comparison to endpoint assays where you receive only one single data point, with the Atlas system, you can generate or you can monitor your cell signal over time continuously. And this has the advantage that you also see the kinetics of your compound effect of your cell response. Now, the very well established methodology behind the system is electrical impedance spectroscopy. So impedance spectroscopy allows to monitor a large variety of effects in cells over time in parallel. Because depending on your readout frequency, which you see here, either cell layer is that barrier integrity or cell death is monitored at the same time. So this means the system allows for a unique richness of information that you can gain from your cells. Now, ready to go recording modes with automated graphing of results is available for ease of use. Furthermore, exporting of raw data and final figures is also possible. Now again, coming back to the HESI compounds from the cardiotox study, we will later on see analysis on whether the cell integrity of cardiac-like cells is impaired or not upon chronic exposure of these compounds. And I'm very much looking forward to that talk and as well to all of the other talks. And I guess now it's time to hand over to my colleague Ellie, who will be introducing our first speaker.
Okay, good morning, good afternoon, everyone, depending on where you are in the world, and a very warm welcome from me. Thanks very much, Sonja, for this very nice introduction. We're delighted to have so many of you here today, and we have a great lineup of speakers bringing together the cardiac safety community in our Research Insights Cardiac Safety Symposium 2023. So without further ado, I'd like to introduce our first speaker, Dr. Mike Curtis. I'm very happy to see that he is here, even if he came in incognito mode by calling himself a name that I didn't recognize. So Mike is from King's College London. He's been researching cardiac arrhythmias and antiarrhythmic drugs for more than 30 years. Some of you may know Mike as editor-in-chief of the Journal of Pharmacological and Toxicological Methods. I know I do. Mike's going to talk today about the use of Translational Therapeutic Index in antiarrhythmic drug discovery. So thanks, Mike. If you would like to share your screen and start talking, that would be great.
There we go. That's better. I'm going to talk to you about a pretty simple idea today, which has really filtered into my thinking over the last few years as we have been developing a new ischemia-activated and highly selective antiarrhythmic drug, and I'll get onto that later on. But one of the big problems in preclinical research, particularly in academia, is the tendency for academics in particular to hype their findings and to move swiftly away from anything negative about something new they're working on. I'm a little bit different from that, which has probably hampered my career. But so I'm going to talk today about the use of the so-called Translational Therapeutic Index in antiarrhythmic drug discovery. So over the years, new drugs, new chemical entities have been championed by discovery scientists, both in academia and in industry. And as I was suggesting a moment ago, clues about safety issues are often downplayed when the efficacy testing is done. So safety pharmacology developed after the tafenidine problem of the 1990s, and very swiftly learnt to try and mitigate the problem of taking forward new drugs that are never going to work by front loading with safety pharmacology studies. But there's always scope for improving efficiency, and for me, efficiency means chucking out the drugs and the pharmacophores that are never going to float because of inbuilt safety issues. So I'd like to suggest that it's possible to facilitate the process of eliminating bad drugs by using the Translational Therapeutic Index. So what is this? It was first mentioned in a paper some years ago. It's not been formally defined, but Muller and Milton said that we use the term to emphasize the need to understand the preliminary translational index as early as possible. And it should be understood that there's a continuum of translational indexes that are refined during the process. And we all understand this. We get a clearer idea about the relationship between the effectiveness and the safety of a drug as we accumulate more data and information. But it may be possible to short circuit this process to a degree by a simple approach of accumulating safety and effectiveness data from the same studies. So the TTI is an animal model variant, if you like, of the therapeutic index. And the upgraded definition I'd like to offer is that it's the ratio between the maximum concentration with no use-limiting adverse drug reactions and the lowest concentration with benefit in a disease model. So data from the same experiment. Now that's the twist, if you like. And I did a Web of Science search, and I only found one paper that had a Translational Therapeutic Index in its title. So this concept hasn't really caught on. It's not captured the attention either of discovery or safety pharmacology scientists, as far as I can see. I mean, maybe it's something that people use and don't publish. I don't know. I've got I can't see my title because the way this thing's set up is there's a whole dialog box at the top. Never mind. So I think it says something like using the Translational Therapeutic Index. So there are some simple ground rules, I think, that could be applied when we use the Translational Therapeutic Index. And the first and the most important is that the safety data that one's generating should be generated in the proof of concept model. So you generate clues about safety and clues about effectiveness in the same experiment with contiguous readout. So this allows threshold concentrations for safety and effectiveness to be compared from a single data set. So, and this, of course, will then obviate the numerous confounds outlined by Muller and Milton, where they're thinking about data from different experiments rather than from single experiments where there are variations in pharmacokinetics and exposure, even worse, species setting and whatnot. So you're lost when you're looking at and this is always a big challenge. And I've done a bit of consulting over the years where people show me data sets from numerous different experiments and models, and then say, well, what do you think? Is this a drug, or is it dangerous? I don't know is the answer. So there's an obvious caveat from all of this, of course, and that is that the Translational Therapeutic Index generated from a single model can't be extrapolated. It is self-contained. And of course, what this means is if the Translational Therapeutic Index derived from experiments in a single model look as if the drug has potential to be developed, then one needs to do lots of additional research. But if the Translational Therapeutic Index is found to be narrow or none, this actually provides us with useful information because this can help us make a swift decision about excluding unfeasible drug candidates. So how do we go about using the TTI then? Is there a rubric which we can apply? So the rubric is simple. Never estimate the Translational Therapeutic Index from separate safety and efficacy data. Generate the TTI from a single model. And if the TTI is small or none, that's an important point to consider halting progression. And I've got some data that I'll show you shortly to illustrate this point. Now, if the TTI is large, then clearly it's reasonable then to proceed with further efficacy and safety testing according to the goals and objectives of the company and the program. So can we validate this rubric then using a set of data from a disease-specific database? So let's take a look at this. I can't get my slide to move on. What's going on here? There we go. So here's an example of a data set generating a TTI. So we've been working on antiarrhythmic drug discovery for a very long time, 30 years or more, seeking a drug to prevent ischemia-induced ventricular fibrillation. And as part of this, this process, we've examined countless standard and novel antiarrhythmic agents, generally finding that things are not sufficiently selective in terms of their effectiveness to warrant further work. So as I mentioned earlier on, that it's standard in academia to hype the effectiveness and somewhat downplay the adverse drug reactions in experiments. I've seen this countless times. It even can happen in industry, and I've seen that too. We've tended to take a different approach, and I'd rather be in a position where I can stop wasting time, money, and effort pursuing something that's never going to work. So here's an example case study. So this used our standard bioassay, our kind of entry-level assay, which is an in vitro coronary artery ligation model using the rat isolated Langendorf preparation. There's a review I published on a massive load of data back in the 1990s. So background, we'd previously shown that verapamil was very effective at preventing ventricular fibrillation in conscious rats, but it had loads of adverse effects, AV conduction problems and blood pressure lowering effects. And of course, we know that it failed clinical trials owing to underdosing, which was done to preclude adverse effects. So a typical problem where a drug doesn't have a good TTI. So verapamil, large database, was a good drug to use as a template drug to explore the perfused rat heart model as a TTI generating bioassay. Because obviously, if you're interested in drug hunting, the entry-level model is not going to be a conscious animal model of disease. You want to start out with something simple. We don't like using single cells. The readout is too nebulous. But with the Rat-Langendorf preparation, we have real ventricular fibrillation induced by ischemia as our primary readout. So this was a study then that Andras Farkas did in my lab in the 90s. And he showed that the study was actually focused on a new drug at the time, abefredil. There was quite a lot of excitement about it until it was found to block cytochrome P, one of these cytochromes involved in drug metabolism and had interactions with every drug under the sun. But the main piece of information from this study focuses on the effects of verapamil. So this is a verapamil concentration response study looking at 5 concentrations from 10 to 600 nanomolar, which is within the bounds of reasonableness in terms of human exposure when the drug was tested. And we can see that verapamil concentration dependently inhibited the occurrence of ischemia-induced ventricular fibrillation, abolishing it at 300 and 600 nanomolar. But it had adverse effects, including, we can see on the right-hand column here, AV block. So I've put a little ring around the 300 nanomolar showing blockade of ventricular fibrillation, but some adverse effects. And we can also see in the same experiments that at an even lower concentration, at 100 nanomolar, we saw increases in coronary flow. So if a drug increases coronary flow in this preparation, it's dilating coronary arteries. And any drug that dilates coronary arteries is going to dilate resistance vessels and lower blood pressure. Every single drug we've looked at has this. If it increases coronary flow, it will do have a blood pressure lowering effect in vivo. So increasing coronary flow is a vasodilator action, and this vasodilator action is going to produce an unacceptable blood pressure lowering. So verapamil had no translational therapeutic index. It had adverse effects at concentrations below those necessary for inhibiting arrhythmias, and that's, it's a powerful lesson. When we published this work, that was part of our focus, that verapamil clearly, if people had done this work 20 years before, we would have seen that there was no chance this drug was ever going to be useful therapeutically. So we generated a similar set of data using looking at sodium channel blocking drugs, or class 1 drugs, mexiletine and lidocaine. So mexiletine was the first drug we formally characterized in terms of its TTI. And we published this a couple of years ago, where we characterized mexiletine's TTI in the rat isolated heart preparation. And we found that the threshold concentration for suppressing ventricular fibrillation was 1 micromolar, but in the same hearts we saw adverse effects. So this is PR interval over time. I won't go into the full details of the complexity of the study. We were changing the potassium concentration, the Krebs, for various reasons at later stages of the experiment, but the PR interval prolongation here with mexiletine is profound and notable. So this is an off-target effect on the AV node. So this means that mexiletine has no translational therapeutic index. So we've conducted further work looking at lidocaine, and the data generated are part of a growing database that we're creating for class 1 antiarrhythmics. And the reason for that is that we're using data to gauge the potential value of a new ischemia-activated prodrug of lidocaine, as I mentioned at the beginning of this presentation, known as OCT 2013. So we published on this last year. So in the Rat-Langendorf prep, we compared OCT 2013, or LNO as it's also known, it's lidocaine N-oxide. We found that the two drugs were equally effective. This is high concentration, 15 micromolar, but this is prevention of ischemia-induced ventricular fibrillation here in the rat isolated perfused heart. But in the same hearts, we noted adverse effects of lidocaine. We see bradycardia on the left, in the red with lidocaine, and we see PR prolongation with lidocaine as well. LNO didn't have these adverse effects. So we did conduct, I wish I could see my title. This is going to be a problem for other presenters. They're not going to be able to see their titles because of the dialog box. So in further work we saw then, we did another study with higher concentrations of LNO and lidocaine, much, much higher. And even at these concentrations, there were no effects on PR or heart rate with LNO, and we can clearly see adverse effects of lidocaine. So what are the implications of this? Well, verapamil and mexiletine clearly lack a translational therapeutic index in the Rat-Langendorf prep. From previous studies in our lab, although we didn't present the work in this way, we did report the adverse effects, but we can infer the same conclusions for a range of different drugs, glibenclamide, tadisomil, and all sorts of other putative antiarrhythmic drugs or drugs that affect the pathological process of ischemia. And, you know, the likelihood of anything that people are looking at in preclinical research making it to become a drug, we know it's very low. So the fact that we can't find a translational therapeutic index in our experiments is as one would expect, really. We're not concerned. We think this is actually an indication that the TTI is a very useful piece of readout. So of the drugs that we have looked at over the years, the ones that have been tested for prevention of sudden cardiac death in humans have failed to translate. Generally, it's interesting actually, generally because of lack of effectiveness owing to selection of very low doses in order to avoid adverse effects. So what we're proposing is that the lack of the translational therapeutic index that's been found in studies where effectiveness and safety are evaluated at the same time is a clear indication that the drug will fail. And that, of course, is useful information. So there are caveats, of course. So if a drug's got a promising translational therapeutic index, and I think more than 30 is minimum for most drugs, certainly if the drug has, if we're looking at torsade de pointe liability, then this can trigger further discovery research. We have to continue researching on drugs that look promising, of course. And that's because a good translational therapeutic index doesn't prove safety, especially if it's generated using the in vitro model like our isolated heart, because obviously we can't rule out effects on other organs. But this is the good thing, a bad translational therapeutic index can't be made better, even when an in vitro disease model is used to generate the translational therapeutic index. But that, of course, is provided that the disease endpoint is a hard endpoint, not an unvalidated surrogate. So we use ischemia-induced ventricular fibrillation as our surrogate for ischemia-induced ventricular fibrillation. And we are reasonably confident that this is a hard piece of readout. So what of the drug that we've been attempting to develop? We have simple but clear evidence from our isolated heart studies that the TTI is yet to be determined, because LNO had no adverse effects up to 60 micromolar. I think the highest concentration we studied in the perfused hearts, and 100 micromolar in some other experiments. And we've conducted incremental IV dose studies in anesthetized rats, and the TTI in the perfused hearts compared with lidocaine gave us a strong clue that we may be onto a winner. So we've got patent protection and are exploring a strategy for first in human studies. So I'd like to acknowledge all the countless past colleagues who generated masses of negative findings, drugs with poor translational therapeutic indexes in our standard assay, Sean Rees through to Kat Wilder. Louise Hesketh has done most of the pivotal work with LNO, and she's continuing to work in my lab instead of me. We've been funded by a range of different people, and our key collaborator at the moment is Biotherics and Stefan Ogrzinski. So thanks for your attention. I hope I haven't gone on too long, and I'm happy to answer some questions.
Okay, Thanks a lot, Mike, for this very interesting talk. I must admit, I didn't know anything about the Translational Therapeutic Index before. We have a couple of questions. The first one is, how does the TTI approach translate or fit into the new ICH E14 S7B guidelines draft, if at all?
Well, it doesn't fit into it because it's not being considered, but I think it will be useful for it to be considered. So if anybody has any traction with the people developing the new guidelines, then perhaps it's something they can bring up.
So at what point in the drug discovery process would you think looking at the TTI is a kind of useful parameter?
Well, if you're developing a drug for any condition, the process could begin with high throughput screening to find a ligand, and then you're going to want to test the drug in a disease model. You may want to play around a little bit and look at the pharmacology, but if you can identify an in vitro disease model, because they're much easier to deal with, such as our Langendorf ligation ventricular fibrillation model, then that would be a perfect point to start collecting information that can be used to generate a TTI. So as early as possible is the answer, because essentially when you're looking for effectiveness, you see signals of poor safety, then this is the red flag. And as soon as you start seeing it, this is when you need to start thinking about, do we want to carry on with the pharmacophore, or should we fiddle around with the molecule to find something that doesn't have the adverse effects? So early, as soon as possible, I think.
Yeah. What other in vitro models or techniques other than the Langendorf art do you know of that can be used to sort of predict the TTI?
Well, it depends what therapeutic area you're working in. We're thinking about major organs here. Obviously, isolated brains, not going to be anything we're going to be wanting to work on, although people do use hippocampal slices and whatnot. So essentially, whatever disease area you're working on, if there is an in vitro model that is used in your therapeutic area, then that would be the model to use to generate your TTI. I'm primarily a heart expert, but obviously isolated blood vessels, if you're interested in developing something to dilate blood vessels. I'm not sure there's much call for that these days. We've got enough antihypertensive medicines. Certainly lung, people do use in vitro lung preps. So that's another example, perhaps.
Okay. Well, if there are no other questions, I think we can say thank you very much, Mike, for this very interesting talk.
Pleasure.
And now we will move on to our next speaker, who is joining us all the way from Australia, Dr. Adam Hill. Adam is head of the Computational Cardiology Research Group at the Victor Chang Cardiac Research Institute and Deputy Director of the Victor Chang Cardiovascular Innovation Centre. Adam is an internationally recognised expert in cardiac physiology, safety pharmacology, and computational cardiology. And a major focus of his research is using in silico modelling and in vitro pharmacological assays to better understand drug-induced and inherited arrhythmias and developing improved preclinical screens for proarrhythmic risk. Adam's going to take us into the clouds today and is going to talk to us about a cloud resource for proarrhythmic risk prediction. Okay, thanks, Adam. I'd love to see that you're already sharing your screen.
Okay, thanks for the introduction, Ali. So, as you said, I'm going to be telling you about our proposal to build a cloud-based resource for proarrhythmic risk prediction. And this is an area that my lab and Jamie's lab have worked quite extensively in from a research point of view. So this talk is really about harnessing that academic progress into something that's potentially commercially useful or of value to industry. The problem we're addressing here is one we're all familiar with, which is that of drug-induced arrhythmias, which really came to the fore in the 1990s when a range of drugs were withdrawn from the market because of their potential to cause sudden cardiac death. And it was identified that in most cases, this side effect was linked to block of the hERG potassium channel in the heart and the associated delay of repolarization and QT prolongation. And once that link had been established, there were guidelines implemented describing required assays for hERG block and repolarization delay. And those are summarized in the S7B and the E14 documents, where S7B in particular outlined requirements for preclinical screening. And last year, the first set of Q&A were published for S7B, covering things like integrated risk assessments and best practice for proarrhythmia models that I'll come back to later. So how have these guidelines impacted drug development? Well, as we know, there's now this growing concern that hERG activity is an overly sensitive predictor of risk that perhaps has prevented compounds that might have favorable clinical characteristics from being further developed. And it's easy to see why when we put some numbers around that. So there's an estimate that maybe 70% of compounds can block hERG at some concentration, and maybe 30% of new chemical entities have been abandoned solely on the hERG activity. And from a financial point of view, it's estimated to cost about $5 billion per annum for these tests. So it's quite a significant cost burden. And these factors, amongst others, motivated the FDA to propose CEPA, which aimed to quantify the actual risk of arrhythmia rather than having a sole focus on hERG activity in order to lower drug development costs, improve characterization of risk, and allow early and informed go or no-go decisions. And in that regard, we all know the CEPA working group laid out an exemplar pipeline from a high level that essentially consists of these three steps, which are high throughput screening against multiple ion channels, so not just hERG anymore, computer simulations and model optimization, and a risk prediction model. And this was all published in a series of papers, which I've referenced at the bottom here, which culminated in this white paper in 2020, which outlined those general principles for risk prediction models that form the basis of some of the S7B Q&A. But each of those stages of the new pipeline actually present quite significant challenges for pharma or contract research or academia. So for the data processing, we're now considering much larger data sets against multiple, so 7 different ion channels, again, not just hERG. The well-known complexity of protocols used to measure hERG binding kinetics and the quality control associated with those assays. From a simulation point of view, we're now talking about complex model parameterization, again associated with those models of drug binding kinetics, a requirement for uncertainty propagation, an extensive simulation. So giving us this requirement for large-scale compute and the associated expertise. And finally, in terms of the risk stratification, there's the identification and calculation of new metrics, and also the site-specific qualification and validation of those for individual research sites. So lots of new hurdles in terms of expertise, capacity, and changing workflows. But this same set of circumstances we thought provide us with a window of opportunity. So to meet this need, we proposed this cloud-based service for assignment of proarrhythmic risk that might facilitate CEPA compliance when necessary. And what do we see this looking like in a cloud workflow? So here's a schematic of a potential pipeline where we've got data streaming from automated patch clamp systems into our cloud platform, and we've been thinking about these tiers of analysis. So at level 1, you've got quality control and maybe simple assays like hERG hits or hERG dose response. At level 2, we're getting a bit more complex. So we're talking about analysis of multi-ion channel data, incorporating that into in silico models to predict repolarization effects. And then this comparison or ranking of test compounds relative to a reference library of drugs of known risk. And finally, level 3, where we might have a qualified risk model running in the cloud to assign specific risk labels. So that's the big picture. But as a first step, we've implemented the FDA's validated pipeline, which was based on this metric QNet as a proof of principle or a minimum viable product to try and demonstrate the cloud concept and test the value for different users in different sectors. So back to that pipeline and what we've done to facilitate and automate each stage. And the first step is in the data handling. Now, this might seem like sort of the more mundane end of the spectrum, but it's really critical in terms of getting good quality data through into the simulations. So this includes streaming of the data set, so the JSONs, the .dats from the SynchroPatch into the cloud, this fully hands-off advanced quality control and the data processing and analysis. And in particular, I think there's a method for robust and consistent quality control is really important. So I'm going to spend a few slides talking about that. To illustrate the problem, here's some data from GSK and from Astra that was summarized in the paper from Gary Miram's lab some years ago, which showing really quite significant variability up to an order of magnitude in IC50s here and here, and even more in the Hill coefficient from measures of potencies of a single drug against hERG at the same site on the same platform. Now, it's really important that we minimize these effects because the uncertainty in the measure of potency then propagates through to the calculation of risk metrics. So while I know there's a lot of factors that can contribute to this, I think one area we can reduce this variability in is by applying really robust quality control. So it's an area we've put quite a lot of effort into in terms of getting it right for our data processing workflow. So in our pipeline, quality controls handled at multiple levels. And in terms of what it looks like, first there's extraction of data and metadata from the raw file. So these are things like plate type, the voltage protocol, but also the current or cell type. And these are important because the downstream quality control are specific to different currents and different plate types. We've then got platform-based quality control, again, which is specific to the plate type. These are conventional parameters that we're used to seeing straight off the machine. And then current-based quality control. So for the sodium channel, we might use rise time. For some potassium channels, we use the morphology of the activation phase, things like current amplitude and stability. And then finally, getting onto pharmacology-based QC, where we're looking at goodness of fit of the Hill equation, whether there's an appropriate concentration range relative to the IC50, and whether we've included or how the negative controls behave. So the first of these is the platform-based QC. As I said, these are the measures straight off the machine that we're, any SynchroPatch user will be familiar with. Things like seal resistance, series resistance, and capacitance. And there's also some derivatives of these, such as delta seal. So how do things change over the course of the experiment? And what you can see here are the distributions of those parameters across a plate, and the red region highlights the range of values that we take through the quality control. And we typically lose maybe 20% of sweeps or 10% of wells on the plate, which I think is quite typical of a SynchroPatch run, as you'd expect for these conventional parameters. Then in terms of extracting data, here's an example of a hERG current. I've visualized the average traces for control and drug, from which we generate these block profiles over successive sweeps for control, for the test drug, and for the maximal block. And you'll see in a few slides' time that we then use this experimental profile for more advanced quality control. A couple of other very similar examples just to illustrate some of those current or cell line based quality controls. So for KV7.1, for example, we look at these amplitude ratios of the current onset to eliminate those currents that have non-sigmoidal activation, such as that one shown here. that might reflect a sort of inappropriate or irregular subunit stoichiometry in that particular cell. Similar for sodium channels, where we might filter on time to peak as a simple way of ruling out poorly clamped large currents that might confound the measure of block. And we've got similar current specific or cell line specific quality controls for most of our cell lines that we're analyzing. Excuse me. As I said, from those block profiles, we can also apply quality controls based on things like rundown, on stability, or on slow block. And we do this by using linear fits to individual portions of the experimental profile that you can see here illustrated with the dashed lines. And then we can exclude experiments based on the slope of these fits, maybe reflecting fast rundown or run up, et cetera, et cetera. And this is actually the stage that's probably responsible for most of the data attrition. And it's the one that coming back to that problem of variability in hill slope really tightens up that value across the plate. So what about some before and afters? Well, here are a couple of relatively extreme examples just to illustrate the point. We've got before QC on the left, post QC on the right. And you can see hopefully that we really do get an improved fit of the dose response curve. And we're able to maximize the data usage across the plate. And critically, we get improved confidence in the estimate of the IC50. So that's illustrated by this red box. And you can see that the width significantly decreases with the onset of quality control. And also improved estimate of the hill slope. So you can see highlighted in the box here, we're going from a very shallow hill slope pre-QC to something that's pretty reasonable after processing. In terms of the final product for the data, obviously this is a nice example, but we've got well-defined dose response curves. We've got tight 95% intervals on the IC50. We've got appropriate hill slopes. And we've also got tight confidence intervals on the dose response curve, so the green shaded area here. And this is important because there's reduced uncertainty in the measure of potency that's then propagated through to the calculation of the risk metric. So the next step is the model optimization and simulation. Now, these sort of simulations are pretty routine stuff in the field normally. What we do is we take the cell model, we alter the conductances of ion channels in that model based on a drug potency measured in vitro, and we simulate the effect on the action potential to derive a risk metric, which in the FDA case is QNet. But for this approach, there's quite a lot of added complexity, both from parameter fitting for the pharmacodynamic model and the propagation of uncertainty in measured block through to the calculated metric. So, for example, for every dose of every drug, so for the CEPA panel, that's 28 drugs, we've got to fit this pharmacodynamic model to 2,000 bootstrap samples. We've got to use Markov chain Monte Carlo to get IC50s and Hill coefficients for all the 7 currents for 2,000 samples. We then simulate action potentials for each drug thousands of times to equilibrate it at multiples of the Cmax in order to calculate the QNet and the risk metric. And what we found, there was a really large compute bottleneck for us associated in particular around the fitting of the pharmacodynamic model. And on top of that, there are very specific hardware, operating system and software library requirements that are necessary to really robustly produce the FDA data, which is also quite a challenge for the user. To get over that, as you guess with our discussion of the cloud, we're using Microsoft Azure. So Azure is a cloud platform which allows applications and services to be developed, built, and deployed. There's large-scale data storage and critically really massively scalable compute on demand. And importantly, for an application like this, where we're thinking about dealing with commercially sensitive data, there's the ability to securely permission and share data. And it also operates with hardware in 140 countries around the world, so we can preserve data residency that might be necessary to maintain compliance in the country of origin. Here's our implementation on Azure. We're using Azure Batch. Critically, again, we've used containers, so we've captured all the code, the operating system, the libraries, all packaged in a Docker image, so we don't have any of those problems with dependencies. We built an API using Azure Functions to deploy those containers onto Azure Batch. This is a scalable compute environment, and we're using Azure Share for storage. You can see here some of the outputs for just one particular drug. Now, these histograms at the bottom show the distributions of the 6 parameters recovered from fitting of this pharmacodynamic model to the 2000 bootstrap samples, and we're comparing Azure on the top to the FDA published parameters on the bottom, and you can see there's a very good matchup. These parameters are then fed into simulations, and what you can see here is the dose dependence of the distributions of QNet for 4 doses of Zimmelite here, just as an illustration. And critically, Azure really allowed us to smash that computational bottleneck. So we're talking about taking hours on Azure versus about a week per drug on our local machines. Obviously, that would depend on the resources available. And again, we've got very accurate reproduction of the published data all the way down to to the level of this kinetic parameters from the model. So finally, the risk metric. All I'm going to show here is just a summary of the calculated Torsade metric for our Azure pipeline for high, intermediate, and low risk drugs in the CEPA test panel, overlaid with the published FDA scores and errors. And you can see that there's very robust reproduction again. So we've got to the point now where we've got the full pipeline implemented in the cloud in Azure. And we're at the stage where we're looking for partners from pharma, CRO, industry to test, discuss, and develop further this process. Now, just before finishing up, I want to touch on one other aspect of that big picture schematic I showed you earlier in the talk, which is this concept of a gold standard reference data set, which we might use an alternative to explicit risk classification. So rather than implementing a risk prediction model to assign labels, what we're doing is developing the capacity to score or rank test compounds relative to a reference library of compounds of known risk. So what you can see here is an illustration of the concept using a new proarrhythmia score that we've recently developed in our lab, which you can read about in this preprint at the bottom here. And what we're doing is working on gathering the gold standard data against all these ion channels and using our data processing and quality control pipeline, processing this data so we can implement this ranking and scoring approach as part of as an option in our service. So just to summarize what I've told you, we've developed a cloud-based service for assignment of proarrhythmic risk. What we've demonstrated is data streaming from automated patch clamp platforms into a secure cloud. It uses hands-off, fully automated advanced quality control. There's automated processing and analysis of the data. Model optimization and risk metric calculation are handled efficiently in the Azure cloud, and we've shown robust reproduction of that FDA-validated approach. I've also discussed this concept of risk scoring relative to a gold standard reference data set as an alternative to explicit risk labels, and I just reiterate that point that we're very much looking for testing, development or beta partners, any feedback, positive or negative, and please feel free to contact either myself or Jamie on the addresses shown here. if you missed this, I'm sure someone from the Nanion team can put you in touch with us. So just to say thank you to the people involved at Victor Chang. Satya Arjunan is the lead developer, who is sort of ably assisted by Jessica Farr. Jamie is obviously a close collaborator of mine in this space. Dave Mihares is a cloud engineer who's been working with the Microsoft team to move all this onto Azure. Rob Love is a business development manager employed by Ashra, who are some of the major funders of this work. Jeff MacArthur is a facility manager at the Victor Chang Innovation Center, who is gathering all the in vitro data for the reference data set. And finally, thank you to Microsoft Azure and to the team at Nanion for your support and for having us here today. So thanks, Ali.
Thanks a lot, Adam. That was a really great talk. It seems like a really useful tool for automated patch stamp users studying safety pharmacology. I have a couple of questions here. Please, if anyone has any other questions, please put them in the chat or raise your hand if you'd like to join in with the discussion. Does the data, the submitted data, require some kind of specific open data format, like similar to that are submitted to the FDA?
Right now, no. We're using the raw .dat and the JSON files from the metadata from the SynchroPatch platform. I think in terms of broadening the utility of this, I know we probably don't want to talk about competitors here, but if, you know, equally the platform might be valuable for people using other platforms. So, you know, in the long run, an open data format would certainly facilitate that. Rather than, you know, making bespoke data ingress pipelines for lots of different data formats.
Yeah, okay. Of course, we have to admit there are other competitors out there that may want to use this tool. People
Not
using
for today.
So what is the data that gets fed in? Is it the raw data, or is it already processed IC50 data?
No, just 100% raw data. Yeah, so far we've done those 7 channels, hERG, 7.1 sodium channel, calcium, late sodium, 4.3 and Kur 2.1. So we're just taking in the raw data from those, and based on the metadata that's on the platform, you know, we're applying the relevant quality controls both for the plate and for the particular current type, and then processing those. Processing those data sets to derive our own IC50s post QC. A challenge, if we can talk about it, is making sure that sufficient metadata is entered, you know, by the SynchroPatch user. It certainly makes the hands-off approach much smoother, much easier if there's a very complete data set that's uploaded on the machine. So, you know, the old adage, you know, you can't account for people. We've always got to be sort of defensive in how we sort of build things to make sure there's, you know, always an option for user input rather than just fully automated platforms.
You then have to sort of recommend certain experimental protocols, like incubation time. Obviously, things like that are quite important for, you know, whether it reaches steady state and things. So you have sort of certain
100%. And the point that I made about, you know, there's a lot more to the data variability than just quality control. There are things like the experimental protocol, the cell line, all those sort of things that are you know, beyond our control. But what we can do is eliminate a lot of it, I think, with really robust and careful QC. And to whatever extent we can, you know, we can recommend appropriate, you know, experimental protocols. And as I showed with those fits to the, you know, rundown slow block, we can at the very least flag those experiments where we don't have equilibration, or we can eliminate them completely. Because for sure, when we talk about that, you know, really broad variability in Hill coefficients in data sets, we see a lot of that. So, you know, slow compounds like, you know, difetylide, those sort of things, If you're getting incomplete block, they're the things that really contribute to really shallow hill slopes. Similar for rundown, things like KB43, where you've got a lot of rundown at higher temperatures. That really contributes to the altered hill slopes. But we can eliminate all those with that advanced quality control. So we're trying to tackle it to the extent that we can.
Okay, great, wonderful talk. Thank you very much, Adam.
Thank you.
In the interest of time, I think we'll move on and on to our final speaker of this session, Fitzwilliam Seibertz, or Will. I think I can almost call you Dr. Will, but not quite yet, right, Will?
Will, yeah, that's right, almost there.
Will is doing his PhD at the University Medical Center Göttingen with Professor Niels Voigt. And Will's been using the synchro patch during his PhD to study stem cell-derived cardiomyocytes and also primary cardiomyocytes at high throughput using automated patch tram. And he's going to present some of his work today in his talk entitled APC in the Atria, an overview of high throughput cardiac chamber-specific electrophysiology. Okay, thanks, Will.
Cool, thanks, Ellie, for that nice introduction. Yeah, exactly right. So I'm the PhD student in a basic science research lab. And the main focus of our group is actually to study the underlying molecular mechanisms of atrial fibrillation, which of course is a highly common cardiac disease, which is unfortunately associated with very clinically poor outcomes for patients. And so I'm going to talk today about how high throughput electrophysiological techniques, such as automated patch clamp, have really benefited our lab in the rapid characterization of atrial specific cellular electrophysiology. So just for a tiny little bit of background, atrial fibrillation is characterized by the loss of function of the sinoatrial node. Now, normally in a healthy patient, this node acts as a pacemaker and ensures steady and smooth propagation of excitation across the atria and then eventually to the ventricles. However, in this disease, the loss of function by the pacemaker is facilitated by the development of very high frequency ectopic beats across the atria, which cause the atria to, of course, contract rapidly and fibrillate. And this constant activity actually stimulates the cardiomyocytes of the atrium to develop different electrophysiological characteristics. And this then, unfortunately, promotes the worsening of the disease. So the cells change, their electrical currents are remodeled, and the disease gets worse and worse. These changes are known as electrical remodeling. And there's a very small example here taken from cardiomyocytes directly from healthy patients and patients with atrial fibrillation. We can really see a decrease in the L-type calcium current density in the atrial fibrillation patients as a result of this remodeling. So when we look at the history of antiarrhythmic drugs and atrial fibrillation treatments, we can actually see that most of the basic science research work into the mechanisms, the cellular or molecular mechanisms of atrial fibrillation has only really taken place in the last 20 years. This is in stark contrast with the vast majority of rhythm control drugs that have been developed to treat AF for patients, which of course have been developed over the last 40, 50 years. And we assume that this difference or discrepancy is based on the fact that these drugs were developed potentially with a very low translational therapeutic index as well. These drugs have been developed more on observational readouts or recordings rather than a deeper understanding of the molecular mechanisms actually going on. And so we believe that currently this discrepancy between the available drugs and the development of new compounds that have gone on and this increase in the interest in studying the mechanisms is due to an incomplete set of models with which we can actually accurately investigate the mechanisms of atrial fibrillation. What I mean by that is that much of the mechanistic insights that we've achieved over the last 20 years has been done by using animal models. And animal models, of course, contain very highly species-specific differences in their electrophysiology and are therefore maybe not so appropriate for translation into human patients. And so I'd like to talk today about an alternative model with which we can study human-specific atrial electrophysiology And by that, I'm talking about atrial subtype specific induced pluripotent stem cell derived cardiomyocytes. So these iPS cell cardiomyocytes, they've been a well-established technology in the last 20 years. They very nicely allow somatic cells from humans to be reprogrammed to display a pluripotent state. And from then on, they can be further differentiated into any major cell type in the human body. For this timeline that I'm showing on my slide here, this is an overview of the differentiation from iPS cells into cardiac cells, or iPSC-derived cardiomyocytes. It's a 20-day process. We involve many different transcription factors and many different constituents that we give to the cells at various time points to really facilitate their development into real cardiomyocytes. They contain contractile machinery, they contain appropriate electrophysiology. They're a very nice model for us to use in the lab. Now, usually it's quite well known that using standard protocols, iPS cell-derived cardiomyocytes contain a very wide arrangement of subtypes. You've got some ventricular types in there, you've got some atrial, you've perhaps got some nodal subtypes. And so in order to really shorten this spectrum and to isolate only atrial subtype specific cardiomyocytes, we're employing a relatively new technique, which simply involves the application of retinoic acid early in the differentiation process. Now, this is a vitamin A derivative. It's been recorded to be increased during embryogenesis when the primitive heart fields are first developing. The fields that eventually form the atria seem to have a higher concentration of retinoic acid in the developing embryo. And so if we actually put this onto our iPS cells early in differentiation, we can actually stimulate the development of a very nicely pure population of atrial iPS cell cardiomyocytes at the end of our differentiation process. Now I'm showing you some nice staining here to illustrate what I mean. In the green stain, we're looking at ventricular markers, in this case, ventricular myosin light chain. And you can see in the cells that remain untreated with this extra addition, we've got a high population of green cells, high population of ventricular cells, very nice. However, once we add the retinoic acid and stain for atrial specific markers, shown here in red, we can really see that a huge population is obvious, showing the atrial isoform of myosin light chain as well. So using this technique, we're reasonably happy to assign these two different cell subtypes as either ventricular subtype specific without the treatment or atrial subtype specific with the treatment. And now, of course, my job in the lab has been to begin our investigation of these atrial cells to probe their electrophysiology to see if they are adequate as an in vitro model for studying AF. And this is, of course, where our very nice automated patch clamp system, the SynchroPatch, comes in. That requires not so much introduction, but Nanion have developed this lovely high throughput device in which cells can be automatically placed by a robotic liquid handler moving across the screen there into the recording module. And this module contains 384 individual channels by which up to 384 cells can be probed for their voltage characteristics or their current characteristics as well. And so in a simple batch of experiments, I split a plate into ventricular iPS cell cardiomyocytes and atrial iPS cell cardiomyocytes and began to investigate their electrophysiological characteristics. And I'll just provide a little run through of what I've been able to do so far. Measuring action potentials, of course, measuring the voltage characteristics, these cells did show us a very nice subtype specific difference in their action potential morphology. Now, this is the same as in human patients. Ventricular cardiomyocytes do tend to give us much longer action potentials with a more obvious plateau there. And that's exactly what we see in these iPS cell cardiomyocytes as well. We see a longer action potential for ventricular iPS cell derived cardiomyocytes and a shorter, more triangular shaped action potential for our atrial cells as well. So this was a very nice first step. These cells obviously have some atrial specific markers, and so far they show us very nice atrial looking action potentials as well. Now, the beauty of an automated system such as the SynchroPatch is that it actually allows for the measurement of many electrophysiological parameters during one experiment. And so for us in the basic research area or basic science area, that's very exciting to cut down on time and squeeze out as much data as we can from minimal cellular material. And so once I change amplifier configuration and change a few other bits and bobs, I start to measure the sodium currents as well. This is just nav 1.5 at the moment. And we see very nice, also very clear subtype specific differences between these two cell lines, which is also exactly what we expect to see in human native or primary cardiomyocytes as well. So we're able to get very nice voltage dependent characteristics here and very nice and expected nav 1.5 currents as well. Moving on, we wanted to really investigate as much as we can to get a full panel of general electrophysiology from these cells. And we also saw very nice subtype specific differences in the L-type calcium current, Cav 1.2 as well. Much larger density in the ventricles and a smaller density in the atrial cells, which is also exactly what we expect to see from or compared to the primary methods of methods of investigation. Now, lastly, or almost lastly, we also wanted to look at the IK1 currents. This is the KER 2.1 channel. And this is very important for these cells to have some kind of stable resting membrane potential. So it's been a hotly debated topic whether these iPS cell-derived cardiomyocytes actually have IK1. And then as the technology has developed over these last few years, it's been relatively clear that indeed IK1 can be robustly measured in these cells. And we can even show that they show subtype specific differences as well, where if we compare the inward component of IK1, what I'm showing here is the current readout during a depolarizing voltage protocol. So we're essentially looking at a real-time IV curve of KER 2.1 here. And we're able to see subtype specific differences here as well. So we've got larger IK1 in the ventricles and smaller IK1 density in our atrial cohort as well. Now, something a bit more exciting is the identification of only atrial specific currents. So far I've just shown a nice panel. Yes, it seems to be smaller in the atria, just like it should be, very nice. However, the atrial or the cells of the atrium also contain atrial subtype specific ionic channels as well. These channels are absent in the ventricular cardiomyocytes and therefore show a very nice overview of, sorry, I'll just turn those monitors off. A very nice overview of the real atrial specificity of our cellular populations. So once we stimulate these atrial specific currents, what I'm talking about here is the acetylcholine activated inward rectifier current. Now this is only shown in the atria of primary models. And it's very cool to be able to see that in our iPS cells, particularly measured with automated patch clamp, once we stimulate these atrial only channels, we see a response, of course, only in our atrial cohort. And that shows an increase in the inward rectifier density upon application of a drug which activates these atrial specific channels. You can see that in the atrial cells, this presented as an increase in current density. However, no change was recorded in the ventricles as well. And so that was really nice to be able to validate our atrial cohort as really containing atrial specific electrophysiological characteristics. Now we feel on our side that the SynchroPatch also gives us a very unique opportunity to use this method of screening for atrial specificity as a simple way to give us an overview of how pure our atrial populations actually are. So in a separate experiment here, what I've done is the same experiment, and I've simply quantified the response to carbachol in both ventricular and atrial preparations. If the cell presents as a red box here on the screen, that indicates that it is showing positive for this IK-ACH atrial specific current. The black box indicates that it's negative. And so we can see that in the atrial cohort, the response to carbachol was up to 78% of recorded cells. And that essentially means that within this cohort here, we've measured a atrial purity, as it were, of 78% and a ventricular, let's say, contamination of 22%. And that's also a really nice kind of elegant screening method that we've started to use in our lab, just as a separate testing method to really validate the atrial subtype specificity of these cells. For the other basic scientists in the audience, at least, we also took these over to our colleagues who did a flow cytometry analysis on only atrial cells, and they actually found quite similar percentage purities for the atrial structural isoforms of myosin light chain as well. So an overview of the same batch revealed an atrial, or let's say atrial positive cellular cohort of 75%, which lines up very nicely with our patch clamp data. So we were very excited about this new addition to our lab as a way to really nail down this atrial phenotype and ensure that we have an atrial specific model that we can use for future measurements. So I also want to quickly highlight, for me as the PhD student, I also want to quickly highlight the amazing, intense increase in data throughput that the SynchroPatch adds to our lab. Because all of these data that I've shown today, all of those little points and different currents and everything like that, that took me one day to achieve, which was after some years before we got the device of me sitting on the manual patch clamp rig. That certainly would not take one day. That would be more like 1 year. And so as an intense endorsement of high throughput systems for validating iPS cell electrophysiology, we couldn't be happier with the SynchroPatch in our lab. We've only had it for maybe just less than a year at the moment. What I've shown you today, that just simply came from one batch of ventricular cells, one batch of atrial cells, and then 2 plates, one for the experiments and one for the screening. So that was a very satisfying process for us to be part of. Now, as a little bit of preliminary data, just to bring this full circle, now that we've validated our atrial cell line, therefore we can actually begin the real study of atrial fibrillation mechanisms. And so the next part of this project involves the high frequency pacing of atrial cells in order to see if we can replicate the electrical remodeling that occurs in patients with AF, or with atrial fibrillation. In order to see if we can replicate that in vitro, we simply tachypace these cells. We have one group exposed to normal frequency or sinus rhythm electrical pacing at 1 Hertz. And then we have another group exposed to 3 Hertz tachypacing, or let's call it the atrial fibrillation group. Now, these preliminary data that I'm about to show, that's happened only after 24 hours of stimulation, but we were able to really robustly see evidence of this electrical remodeling, so-called atrial fibrillation associated electrical remodeling in our atrial cohort, where we did see this characteristic reduction in L-type calcium current only in the 3 Hertz paced group, or in the atrial fibrillation group. And that's really exciting. This is still preliminary data at the moment, but this means that we really have a robust model which replicates the very specific cellular conditions that are experienced by atrial fibrillation patients, and therefore gives us a very robust model to be able to investigate these mechanisms further, and of course develop future potential therapeutic targets or treatments as well. So that's where this project's heading, and that's an example of how the SynchroPatch can be implemented in a more basic science or academic setting in order to further some, hopefully soon, future generation of antiarrhythmic drugs as well. That was probably a little bit short there, but I'd like to end it there, and thank you all for your attention. A big thank you to my group, my boss, Professor Niels Vogt, and the other groups around the Göttingen campus who have helped us out with various other aspects of developing these cells and testing these cells. And of course, our very, very deep gratitude to Nanion, who have helped us immensely in getting this fantastic device up and running. So with that, I'd like to stop there and I welcome any more questions. Thank you.
Thank you so much, Will. That was a wonderful talk. Really nice validation of your atrial cells. We have a couple of questions, and I see that Adam has his hand up. So I will go to Adam first of all, if you'd like to ask your question.
Yeah, thanks, Will, for a lovely study. I know how hard those experiments are, so congratulations. I've got a few real quick questions you can just answer with sort of one-word answers if you want. But what sort of success rates are you getting for your atrial versus ventricular cardiomyocytes on the synchro patch?
Yeah, so I can answer that pretty quickly. We're getting absolute success rates for the actual currents between 30% and 10% for the overall cohorts. Ventricular cells are more successful than the atrial at the moment. We're seeing if we have to differentiate for ventricular atrial, we're probably looking for a sodium, let's say, we're probably seeing around 30% success for ventricular and around 25% for atrial. So we're always a little bit of a step down in the atrial category. But overall, we're around the 30% mark.
And doing anything special in the dissociation, or what enzyme are you using?
We're actually, I haven't told Nanion this, but we're actually ignoring a lot of their nice recommendations. We're using really harsh trypsin enzyme-based digestion processes. I can see Ali rolling her eyes there. I haven't had too much success with gentle digestion procedures. I'm not sure exactly why. And so in the chase for just speeding things up, I've gone back to our usual trypsin-based digestion, and it does seem to be working reasonably well to give us those success rates. I'm sure with further optimization, we can get that much higher as well.
Yeah, I think our experience probably correlates with that. We ended up back at trypsin. One more real quick one then. With your tachy pacing, do you see any overt arrhythmias in any cells, or just the remodeling?
At the moment, we actually just see the remodeling. So we don't see any clear arrhythmias such as, or triggers such as after early depolarizations or delayed after depolarizations. We simply see the maintenance of a higher spontaneous rate after the pacing.
Yep.
And the remodeling has been our major finding so far, as a sense for some kind of arrhythmic phenotype.
Okay, great. Thank you. Great talk.
Thank you.
Okay, thanks, Adam. Thanks, Will. We have a couple more questions. I have one from Marcus, my colleague. He says, fantastic data to see. Thanks so much. Are you planning to differentiate iPSCs to pacemaker cells?
That's a good question. And from my side, from our side, no. So we currently, there are protocols out there, but we have very little experience in generating specialized protocols to only produce nodal cells at the end. So from our side, unfortunately, we won't be doing that anytime soon.
And from Jamie Vandenberg, we have great work. Will, in addition to APD90, did you measure the resting membrane potential? And if so, was the average value less negative in atrial compared to ventricular myocytes, consistent with the differences in IK1?
That's a very good question. At the moment, with the synchro patch, we are using quite high holding currents to get these cells at a hyperpolarized enough level to initiate an action potential. And so based on the data that I've created here, we saw no difference in resting membrane potential between the two subtypes. However, I didn't measure absolute resting levels in the absence of holding currents. So when we, using other models, when we put these cells into three-dimensional tissues, we do see changes in resting membrane potential consistent with the IK1 values. So outside of the automated system, we do see a generally more depolarized resting membrane potential in atrial cells. However, not in the data I showed today.
Another one we have from Bogdan. Did the atrial or ventricular stem cell-derived cardiomyocytes feature spontaneous pacing in culture?
They did, yes. So in the absence of any other intervention, so we weren't culturing them under any special conditions, they showed spontaneous beating that was consistent with other literature. So we saw quite high frequency spontaneous beating in atrial compared to ventricular. However, both were below 1 Hertz in terms of their basal spontaneous rate. So atrial, we saw around 0.9, 0.8 Hertz spontaneous beating rate, and ventricular were a lot lower, around 0.5 Hertz spontaneous beating rate.
Okay, and another one from Shinsuke-san. What's the mechanism of LTCC current decrease?
That's a very good question. So it's actually a calcium-dependent mechanism. And what we're seeing is that once you're stimulating these cells so much, they're experiencing a huge amount of calcium influx during their rapid contraction relaxation system. And this actually activates an NFAT-dependent signaling pathway, which downregulates L-type calcium channels in the membrane. We also can see, or others before us have also seen, that it's also a phosphorylation-dependent process. So as well as L-type calcium channel downregulation or trafficking away from the membrane, we're also seeing a decrease in phosphorylation of these channels. And decreased phosphorylation means we see less current coming through. I've just the other week, I also tried putting a phosphatase inhibitor on these cells. So we wanted to increase the phosphorylation. And we saw an almost reversal of this electrical remodeling. So we saw the L-type calcium current get bigger and back down to normal levels. So there's a bunch of different mechanisms in play, some involving downregulation of the actual channel and some involving phosphorylation-dependent mechanisms as well.
Hey, great. Thank you so much. Are there any more questions? I don't see any. So, okay, well, thank you, Will, and thanks to all of our speakers from this morning's session. This was a great mixture of different aspects of cardiac research.
Hello, everyone. I think you can all hear me. I think you can all see me. Welcome to session 2 of this year's cardiac safety meeting. My name is Elena, and I will represent Nanion this afternoon or morning, depending on where you are at the moment. I'm very happy and excited to welcome 4 excellent speakers this afternoon. And as one, our first speaker's title starts with single step to a staircase, we're building this staircase today in our talks, and we will have various topics. We'll start from investigations of cardiac ion channels, and then we will continue up to updates on the chronic drug-induced cardiotoxicity assessments from the hERG cell working group. Very interesting talks to come, and I think Monique Windley from Down Under is here with us. Thank you very much, Monique, for joining this late. Monique is a senior postdoctoral scientist at Victor Chang Cardiac Research Institute, and she will be the first one to go up that staircase today and introduce us to high throughput protocols for measuring hERG drug block. Monique, are you there?
Yes, I am.
Oh, perfect. Thank you.
Can everyone see my slides?
Yes.
Okay, thank you very much for your kind introduction, Eleanor. And I'll jump right in. Okay, so as many of you will already be familiar with, the role of hERG drug block in the generation of cardiac arrhythmias is crucial, such that hERG drug block reduces the IKR current in the cardiac tissue, and this results in delayed repolarization of the cardiac ventricular action potential, which can result in the generation of early after depolarization, which puts a patient at risk for further development of fatal cardiac arrhythmia to SARD depend. And this is evident in the electrocardiogram waveform as a prolongation of the QT interval. And as a result of this strong link, in 2005, the guidelines for preclinical testing were changed such that drugs were required to be evaluated for hERG drug block as a surrogate for proarrhythmic risk. But it's since been discovered that not all hERG drug block results in arrhythmia or even action potential prolongation. So as a result of this, the comprehensive in vitro proarrhythmic assay was developed, which is a major collaboration internationally, which was designed to develop a new mechanism-based test for proarrhythmic risk. This was set in a number of different stages, such that initially to test the effects on a range of different cardiac currents using the gold standard patch clamp technique, looking at potency, and in the case of hERG, also kinetics. And these values are then used to inform in silico cellular simulations of the ventricular action potential, which can then give us a proarrhythmic risk score. And the SIPR-dynamic protocol was developed to measure the kinetics of block and potency simultaneously in hERG channel currents. And this is the simple step protocol that was developed by the Ion Channel Working Group, which is based on the Milnes protocol. And this was applied a number of times under control and drug conditions. And the initial response under drug conditions is the onset of drug block. Shown in panel C here are the typical responses for control conditions in dark blue, and pink, the onset of drug block, which can be used to derive measurements of the kinetics, and also the final drug block in light blue. And so, as I mentioned, the percentage block over time can be calculated from the drug onset response, which is corrected for the control current. So we have a nice trace here representing percentage block over time, and this is fit with a single exponential curve from which we can derive a time constant value, or a tau on. The initial gold standard data set where we assess the 12 SIPR test panel of drugs with known various risks from high, low, and intermediate was published in this study. And so the next step was to determine the feasibility of this SIPR dynamic protocol in a high throughput system, which is relevant for industry screening methodology. So we used the SynchroPatch 384PE, where we were able to look at 384 recordings in parallel. We chose 4 different drugs from the original test panels from the manual patch clamp study, and these were selected for our ability to accurately measure the kinetics of drug block. And these were each tested at 3 different concentrations. And so to automate the quality control, we developed in-house quality control pipeline, and we analyzed a number of parameters, including the quality of the patch or cell, including seal, series resistance, and capacitance. We then looked at various parameters of the hERG channel current itself, the amplitude of the current, and the stability over time. And then we looked at percentage block and curve fitting, the actual analysis of the data to determine that the analysis was of high quality. And another important aspect of achieving high quality data sets is the isolating of pure hERG. So we needed to automate the leak correction. Now, in comparison to manual patch clamp techniques, the automated patch clamp system is a little different in its setup. So we have a planar patch configuration here where the cell is adhered to the bottom of the plate. And this small hole here is where we gain access to the internal contents of the cell when we go whole cell. And the leak between the edge of this hole and the cell membrane is the leak current. And in planar patch clamp technique, only once there was the addition of this fluoride in the internal solution, which was able to complex with the calcium, was the seal quality consistently being able to maintain to improve throughput and quality of these data sets. So initially we used a minus 90 millivolt leak step, which is found just prior to the test pulse, where we see the current, and this measures only the leak. However, we found that the protocol previously set up was not sufficient for our leak correction. So we were able to incorporate the initial portion of the 0 millivolt step, incorporate data from there where there's no hERG current yet present, to improve the leak correction. So here in black, we have uncorrected data, and purple, we have the incorporation of the minus 90 and the 0 millivolt, where we have good correction of the data, whereas without that 0 millivolt data, we see under correction here. And this is particularly important when we do the subtraction to get the percentage block over time, where we have a significant degree of noise at the beginning here if it's not adequately leak corrected. And there's also changes in the kinetics and the full amount of block measured. So our next step was to then go on to look at the number of sweep repetitions. So in our initial manual patch study, we looked at 5 sweeps, but another study where they looked at 10 sweeps for the physiological temperature data set. So we wanted to compare both. So here we have the percentage data retained while after we've applied the sequence of quality controls. And for the 5 sweep data sets, we have 21% data remaining after we've applied these quality controls. However, when we did a 10 sweep data sets, this was significantly reduced to 9.4%. And this is largely to do with also the length of the protocols that we used. So with the drug block concentrations, where we had a faster onset of block, we were able to use short 10-second protocols. However, where the onset of block was much slower, we were required to use longer 40-second protocols. And this drastically changed or increased the length of the experiments, particularly for those 10 sweep experiments. And this is definitely reflected in the data retained. If I draw your attention to the pink dashed line here, where we have almost no data retained in comparison to the other data sets. So the longer experiments definitely lead to a greater instability and loss of data. But what we found when we compared the results of potency and kinetics of block between 5 sweeps and 10 sweeps, there was no significant differences at any drug concentration. So here we are comparing the 5 sweep data sets between the initial gold standard manual patch data set and the high throughput syncopatch data set. And the black dotted line down the center is representative of no change. And what we see for the potency data, that most of the data lies within less than 2-fold change. However, the kinetic data was slightly more variable, with some of the data sets lying between the 2- and 3-fold range there. So in most cases, the data sets are comparable, although there are significant differences for some. and we explored and show these here for percentage block and the time constant of block. And we believe that these are from a number of different sources. Firstly, related to the high throughput platform itself, and not just the Syncropatch, it would be relevant to any other high throughput platform, that they tend to run at 3 to 4 degrees higher temperature than we can achieve with a manual throughput system. And there is a certain amount of plastic exposure that's required within this high throughput system, although it has been significantly minimized. And for drugs like cisapride and tafenidine, this is a significant known issue. And also the patch clamp solutions are different between manual and high throughput, which I will get into a little bit later. And also we have potential causes related to the protocol length itself. So as we showed in our initial manual patch study, the length of the protocol is quite important. Here we have an example of 30 nanomolar cisapride with a short 10-second protocol. And when we do this subtraction of the data to look at the percentage block over time, we essentially have a straight line. We fit an exponential curve to this, and the results that we get from cell to cell are extremely variable, shown in panel B. However, as we increase the length of this sweep and we start to record the plateau of this block, then we have a very tight data set where we can accurately determine the kinetics of block. And this is also carried across to the high throughput system, and perhaps even more so. Here is a figure where we look at the sweep length divided by the time constant value and the coefficient of variation on the x-axis. And you can see that where the sweep length is less than 3 times the time constant, that these values are quite variable. So longer sweep durations are necessary in these instances. However, what we did find when we compared those results with appropriate sweep lengths for drugs with faster kinetics, that we could achieve a quality control pass rate of almost 50%. So we also wanted to look at whether there were potentially other protocols that we could look at the kinetics of drug blocks. So while spending a lot of time on this SIPA dynamic protocol, perhaps there's other ways to assess. So we wanted to look at if there was some way that we could use in silico hERG binding models that are informed and directly fit to the patch clamp data sets. And then use this to inform in silico cellular simulations and develop the proarrhythmic risk score. So using these information-rich protocols, we can improve the measurement of, or show improved measurement of drug block kinetics and potency by also including drug state dependency information and the effects of drugs on hERG gating kinetics. And the sine wave protocol is one such example of a information-rich protocol, which was designed to understand hERG gating kinetics. And it allows for rapid collection of high-quality data sets over just an 8-second period. And you can see below the current trace from that protocol. And using a simple 4-state channel state model in a simple circular configuration, this model was directly fit to the data set. And this, you can essentially see the state occupancy of each of the various states over the time course of the protocol as it's applied here. And ultimately, this protocol produces a more predictive model than other approaches. And this was, and here we see a single step kind of representative that would be similar to a profile that we would see on just the using the SIPA dynamic protocol for comparison. And so this sine wave protocol was then developed for a high throughput alternative, the staircase protocol, whereas this 15-second staircase protocol, where a number of 500 millisecond steps over a range of voltages were used to observe characteristic hERG decay. And we envision potential applications for drug binding and kinetics for this protocol, where we can directly fit the data sets and improve our models of drug block. So similarly to the hERG dynamic protocol, we applied a number of different sweeps under control and drug response conditions. And we were also able to implement some variations to the protocols, where we can change the staircase duration and the interpulse interval, and even slightly change the protocols themselves. This example is called the staircase within a staircase. And it looks at a different channel state occupancy profile, which may be important when assessing drug block. So, as I said previously, to get good fit of the data, we need to have isolation of a pure hERG current. So here we incorporated the subtraction of endogenous currents by looking at full drug block at the end of experiments, and subtracting the leak current, where we apply, as previously, a single leak step or a ramp to then estimate the leak for the rest of the protocol where we have hERG current present. And an important assumption in this, this subtraction is that the leak current is linear with respect to voltage. Unfortunately, what's been discovered is that the inclusion of this fluoride and calcium complexing causes a nonlinear leak current. And it's been shown that cells on the sinker patch, as you increase the voltage, that it diverts from this linear relationship. So we then don't have an adequate measure of the leak current where we actually have hERG currents present. So we went forward to compare fluoride-free experiments on the sinker patch to standard fluoride-containing experiments. And what we found was, as expected, the seal resistance was lower. And we also had a bit of difficulty obtaining good access to the cells, so they weren't converting to the whole cell configuration. And upon further consultation, we included the cell membrane perforator Essen at 10 micromolar. Wasn't completely sufficient to aid in breaking through the membrane, so we still had a significant number of cells with no current. However, with increased concentrations, we were able to, at least those cells that were able to seal, that had current and were stable. So we proceeded with using the 15 micromolar concentration for further experiments. Now, at this stage, we don't have modeled data fit directly to by any models. So this is just a manual measurements of the percentage block at a single point towards the end of the protocol. So for these three drugs, we found relatively little difference between using fluoride or fluoride-free containing solutions. However, for cisapride, quinidine, and verapamil, we saw significant differences to both the kinetics of block and the degree of block. And these responses appear to be quite drug specific, but we're still getting to the bottom of why this might be the case. We also looked at various modifications of the protocol, increasing protocol length for cisapride, difetilide, and tafenidine. This seems to be quite important as these are slower blockers. We then looked at interpulse interval while the channels are closed, between sweeps. And when we decreased this closure period, we didn't really see a whole lot of effect on the kinetics and percentage block, but that does have the potential to explore various closed inactive state block and dissociation of drugs from the channel while that's closed. And the protocol state preference was explored with the staircase in a staircase protocol. And again, for these particular drugs, we didn't see a whole lot of difference. We also looked at temperature. So previously, all of these experiments have been done at ambient temperature. When we increased this to 37 degrees, similarly as to what we've shown with the SIPA dynamic protocol, there's faster degree onset of block and some changes in the degree of block. And this is very much drug dependent. So just showing an overlay of all the different protocols and conditions that we've tried. There's no consistency between the different drugs, so they're all quite drug specific, and this will be important when we're designing our models of drug block. So based on the simple 4-state hERG kinetic model, which has been already used to inform which has been informed by this staircase in this the previous study, and we envision adding a drug binding kinetic transition to this, which may be quite simple to the open state or to various inactive state. Also allowing us to incorporate drug trapping, as in the SIPA model used, and also in various other orientations and configurations, and even incorporating an encounter complex. So finally, I'd just like to thank a number of people who were instrumental to this study, including Dr. Adam Hill, who's head of the computational cardiology laboratory in the Victor Chang, Jessica Farr, who did the quality control software development, close collaborations with Jamie Vandenberg, and head of the cellular facility in the Innovation Centre, Geoffrey MacArthur, as well as our collaborators in the UK, Gary Mirams and University of Macau, Dr. John Lockley, who will be doing the modelling, drug modelling for the staircase protocol. And thank you very much to the organisers for allowing me the opportunity to speak. Thanks.
Thank you very much, Monique. I guess everyone can hear me now. This was a very nice comprehensive talk from your side. Thank you so much. We have a few questions and they keep coming, so I'll just read them through here. From Ron, what are your thoughts on potential mechanisms of the compound-specific sensitivity, susceptibility to fluoride positive relative to fluoride free you showed?
So we have a number of theories that we're exploring at the moment, where considering whether it may be a solubility issue, that if once the drug crosses the membrane and it's exposed to the fluoride, it perhaps precipitates out of solution. That's probably the easiest solution, but it may be related to an interaction of fluoride with the channel itself, interaction of fluoride with the drug, and a little bit more difficult to explore, but we will be looking into that, potential shielding effects, but at this stage, these are just guesses that we'll be looking into.
And just from my side, I'm a little bit curious, have you used the standard 384 plates or fluoride-free, the new ones that we have?
For our fluoride-free experiments, we're using your specialized fluoride-free plates and solutions.
And here we have Bogdan. He asked, how would the hERG block kinetics data obtained with the Milnes protocol compare with those obtained with an envelope of TS protocol?
Okay. So we did actually start with a envelope of Tails protocol when we were deciding what protocol to go with for this kinetics. And what we found was because of the ability of some drugs to dissociate while the channel's closed between subsequent sweeps, we weren't getting a true representation of the kinetics of block. So you'd potentially be getting drug washing out in between sweeps and then starting all over again. But to get a true representation of the kinetics, you really need to be measuring it while the channels are open, and we're actually able to measure the response directly.
Any other questions? Maybe one more for me. We still have 1 minute. You mentioned the recordings you've done with the temperature variations. I suppose you're working on room temperature. Have you tried it under physiological conditions as well?
So we've only tried the standard fluoride containing plates and solutions. Well, we have actually tried the fluoride free, but at this stage, they were largely unsuccessful.
Okay, this was about the temperature. So
one of the slides, the latest slides, I showed a comparison between the staircase protocol with physiological temperature and room temperature. So we have had a look at that.
Good.
And for the Seibertz protocol. Yeah.
Thank you very much, Monique. Thank you for staying up that late for us. And joining us.
Thank you for listening.
You're welcome. And now we'll go from Australia to Germany. We welcome Bettina Lickiss. She is a very experienced scientist in cardiac field of iPSC-derived cardiomyocytes. She is scientific communications manager and business development manager at innoVitro. And now she will switch gears to a little bit different topic, and she will introduce us into the ways of assessing contractile force of human iPSC-derived cells. Bettina, welcome. You can start sharing your screen. Excellent.
Excellent. Already, hopefully already do.
Very good. Excellent.
Yes. Going into presentation mode. Give me 1 second. Set up. So thank you very much, Elena, for the kind introduction and also the invitation to speak here today. I'm Bettina Lickiss, as you said. I'm business development manager of innoVitro. We're a partner company for Nanion Technologies, and we provide FLEXcyte related services, especially for the cardiac safety field. So quite a well fit here today. And I always start my presentations with a video where you can see the cardiomyocytes beating nicely because it's such a great eye catcher. But today I thought I'd make it a bit more interactive and actually make a little quiz out of it. So here you can see 4 of the wells of the FLEXcyte plate, and you, if you like to, can assign the compounds that you can see here on the left-hand and right-hand side to the respective wells. We also have the FLEXcyte signal underneath it to give you a little bit more of a hint. Put it in the chat box or tell me later. And of course, you will not get the results right now because I'm going to show you a little bit more about the FLEXcyte applications that I want to introduce to you. The analysis of hiPSC-derived cell types, contractile properties, and after that, you will get the results. So stepping into the table of contents, because most of the projects I'm going to show you will be dealing with the technology, I'm going to show you a bit more about the FLEXcyte first, how it works and the parameters and etc. And then we dive directly into the robustness analysis of commercial human iPSC-derived cardiomyocytes, because we get that a lot from clients that they're a little bit worried when they have to switch between lots. So they were not, is it right? Is it going to be changing the results, etc.? So we basically did an analysis of 10 different lots to see how robust they were. Then we're going to swap from this mixed culture cardiomyocytes to a different setup where we look at chamber-specific cardiomyocytes. So cardiomyocyte subtypes, atrial versus ventricular, do a contractile analysis of these cell types, compare them a little bit, also do a pre and post compound analysis, And then we step away from cardiomyocytes for a moment and look into a smooth muscle cell maturation protocol that we established for, of course, also for using it for FLEXcyte analysis or contractile analysis. But because we use the primary cell, we have to work on the maturation first. So I'll tell you a little bit about that. And then last but not least, we were able to have the new Atlas device in our in-house lab, and we're a beta testing site and got some really good data that we would like to present to you. And after that, you will get the results of the quiz I just presented to you. So starting with the technology, or basically why is it so important to have a physiological relationship for the cells? If you look at what we have normally in the usual lab, we have very, regarding mechanosensitivity, we have actually very extreme versions that we provide for the cells. On one hand, we have the regular cell culture plate, so very stiff material. The cells on top cannot really contract. They try, but they are basically too stiff to contract properly. So we have a maximum of stress here. On the other hand, we have usually a lot of cells in suspension culture, so the cells are floating and have nothing to hold on to. So we create a different kind of extreme for the cells, which they're usually not used to. And so they can contract easily, but they have problems relaxing anymore properly. So a different kind of stress, basically. So what we have tried with the FLEXcyte technology, with creating the membranes where the cells are on, basically from the video that you have seen on the first slide, is we create this physiological relationship that we need, that we also experience, that the cells would experience in the body, right? So with this physiological relationship, they have something to hold on to, but they also have the flexibility to contract properly. And this is something we have shown in presentations before already. This has a positive effect on the maturity level of especially human iPSC-derived cardiomyocytes. We get proper positive inotropic effects, etc., etc. Won't go too much into that, but very good for the cardiac safety community. So the technology here, this is one of my favorite videos because you can actually see the membranes very well. The membranes are of ultra-thin silicon substrate, basically, and they reflect the physiological conditions, as I mentioned. You can also see the difference here to the electrodes. The electrodes are actually not part of the plate, they're part of the device. And when the cells are on top of the membrane and they are contracting, you can see that they lift the membranes upwards and therefore create a distance change between the electrode and the membrane. And this is then measured via non-invasive capacitive distance sensing. So we have a label-free way to analyze contractility. And we also do it in a cell monitoring way. So we can do it acutely, but we also go up to chronic several days and measure it. And there's all the data we basically get is then analyzed quickly with a specialized software. Regarding the parameters, we of course start with one of the most important ones, the amplitude of contraction force. So we have a proper force measurement that we can perform here. We have also the possibility to go and measure area under curve, that we can dissect in upstroke and downstroke area under curve, because there are compounds that rather work on the contractile part or on the relaxation part, and therefore it's necessary to dissect these things. We can check the beat rate as well as the regularity or the irregularities, like the early after contractions that you can see here highlighted in red. So this is basically like a downstream physiological effect of an early after depolarization that would happen. And then we also look at the slopes, upstroke and downstroke wise, as well as the duration of the contraction, of course. And then here you can have any kind of peak width duration you want. So in total, we're able to really dissect the contraction into its bits and pieces with the amount of parameters we can measure. I always love looking at the raw data as well, because it just shows you the arrhythmia so nicely. So here we just have a simple control condition of one sweep. So basically a measurement of 30 seconds. And once you apply compounds that have an effect on the contractility, you see that we can see like beat rate variations, like highlighted in red here. We can see the earlier mentions early after contractions happening, highlighted here and in with the red arrows, as well as proper arrhythmia events highlighted with the red arrows down here when, for example, nilotinib is applied to the cells. Right, we got a lot of knowledge about the technology now, so let's dive straight into the analysis of commercial human iPSC-derived cardiomyocytes. I already told you that the background of the experiments was basically to see how robust the different lots would react towards compounds, but also just generally speaking, pre-compound condition-wise. So what we did is we chose 10 cell lots that were produced in a time frame of 2 years. So not only like within a few weeks, but also like a quite long time span. And then we did a pre-compound analysis where we looked at the beat rate, and also did post-compound analysis where we checked nifedipine, so calcium antagonist, as well as sotalol, a hERG blocker. And starting here with the pre-compound beat rate, you can see very nicely that we have very stable conditions of the 10 different lots, with a beat rate ranging from 49 up to 53 roughly beats per minute. So we had a good start, and we then applied the compounds to the wells, starting with nifedipine. And you can see here that we have a concentration-dependent decrease of the amplitude upon nifedipine treatment, with high concentrations highlighted in pink and the medium concentration of 10 nanomolar highlighted in green. So the upper and lower lines here is actually the standard deviations that should be dashed lines, but the presentation won't kind of smudged it a little bit, sorry for that. But it actually shows that the standard deviation is below 10%, which is what we wanted to see, right? That there's a low variation, that this is a robust cell type that we can use for doing contractile property analysis properly. And the same was true for sotalol when we applied it to the cells. We have a very low standard deviation, again, below 10%, and we have basically no effect on the amplitude, but a concentration-dependent effect on the contraction prolongation, which is not really well visible on this slide, but then we try to depict it differently by plotting the different concentrations right next to each other also. And also this is just basically a downstroke duration shortcut. So here you can see that we have a concentration-dependent duration prolongation, which is basically representing the contractile response to the QT prolongation. And on the left-hand side, we have a similar effect of nifedipine, but the other way around, where we see the concentration-dependent shortening of the beat duration with a higher concentration shortening it. So in all, very nice data showing that there's actually no stress about different lots. You can use them all easily when you go for a commercial cardiomyocyte. Next, as I mentioned, we looked at the chamber-specific cardiomyocytes. This is from a different commercial vendor. We treated them basically completely different, completely the same way. We added a 50K per well for both cell types. We had the same medium, did a pre-culture step of roughly 6 days, which is our very usual pre-culture step, and then did similar to the project before, like a pre and post compound analysis. In this case, we use carbachol and aspa. And here you basically have the pre-compound condition of the amplitude of the different cell types. And you can see already with your immediately the different shape reflecting to the different cell type, with a typical ventricular shape with the shoulder and the atrial shape here. Looking at the results of carbachol, which is an acetylcholine receptor agonist, so working predominantly atrial. So we were kind of expecting a strong effect on the atrial cells, which we also saw. Where you can see here that we have a duration prolongation, a strong one, a concentration dependent one as well, from 1 micromolar carbachol up to 100 micromolar carbachol. And for ventricular cells, we see a similar effect, but just much smaller in comparison to the atrial ones. Another compound was tested, was the aspa one on ventricular and atrial ones, where we had the control condition here. And what we can see here very significantly is just 1 minute after applying the compound, which is an L-type calcium channel agonist. So we have a strong increase in the duration for both cell types. And what then is very interesting to see is that after 10 minutes, we have a very physiological effect of aspa on ventricular cells, where we see an increase in beat rate. So all in all as well, this was a nice project to show the differences of the different subtypes of the ventricular and atrial cardiomyocytes using contractile property analysis. Okay,
Leaving cardiomyocytes aside for a moment, we go to smooth muscle cells on the FLEXcyte 96. And the next cell type we basically want to evaluate, or are evaluating right now. This is like the first few projects that we did for it. And because we don't want to go straight with commercial cells, we were using a primary human aortic cell line. The advantage is obviously that we can just grow it in-house and have up to 15 population doublings. The disadvantage is that because they're proliferative, they're also kind of non-contractile, so we had to work on a maturation protocol first. And that's what we did. We actually fiddled about with quite a few different setups and was always like kind of having them in culture for 7 days before we then plated them onto the FLEXcyte plate and had half of the plate with maintenance medium and the other half of the plate in like changing maturation style medium, looking for which one would be the best one for doing a functional, for the readout as having a functional analysis with the FLEXcyte, as well as the phenotypical analysis where we did some immunostainings. And the ones that worked best actually for us was where we had basically a reduction of everything in the medium, where we would go from 5% FCS down to 0.5% FCS, and also basically got rid of the growth factors completely from EGF and FGF. And that seemed to work out very well for the maturation of the smooth muscle cells, as you can see here with the phenotypical analysis. So on the right-hand side, we have the maturation medium, and the phalloidine staining, basically a staining for actin filaments, shows actually very nicely that we have an organized cytoskeleton in comparison to the maintenance medium here on the left-hand side, where everything seems to be still a little bit disorganized. So that was a good step forwards. And then we also performed some functional analysis where we used phenylephrine, so an adrenoreceptor agonist for checking basically the contraction behavior. And you can see here very nicely that we basically, with the blue bars, we have no response in the maintenance medium of the smooth muscle cells that were in the maintenance medium, but the ones that were in the maturation medium did actually react to phenylephrine quite nicely with a almost 30% increase in force of contraction after the maturation process. So here, I think we found a good way of getting our protocol into place for doing follow-up studies, taking more compounds in and checking the smooth muscle cells more regarding their contractile properties. Right, our last project actually is the new product of Nanion Technologies. So that's what Sonja introduced in the morning. We were happy to be beta test site for the Atlas, and were able to have to create some data very quickly thanks to the amounts of wells that you can basically analyze at the same time. Because as Sonja mentioned already, we have the possibility to actually test 96 well plates at the same time. And this has nothing to do with contractility, by the way. This is an impedance-based system, and the frequency spectrum here is actually quite broad. There's a lot of things you can analyze between less than 1 kilohertz and up to more than 40 kilohertz. So just to pick a few, there's the teal mode, for example, interesting for hepatocyte analysis, and cell monitoring mode. That's what we did for our cardiotox study, and cytolysis mode, for example. So the study design was to test chronically the cardiotoxicity for 3 days using the cell monitoring mode. And because there's so many wells and there would actually burn quite a lot of commercial cells, we decided to go for a cell line again and use the H9C2 cell line, so red heart myoblast cell line. That has like acts similarly to the smooth muscle cell line that we use. So they're proliferative, which is great because we can increase them quickly, but also they're rather immature. So we had to use serum salvation again to induce the myotubes. But we were successful with doing that, so that was actually no problem. Here's the myotube, for example, and this cutout here shows that we even have multiple nuclei. And therefore, we were basically ready to go, plated them on 6 96 well plates, and basically performed this study within a week only. That was quite cool. And the compounds that we selected were actually a subset of the HESI cardiotox study that was just being performed. And that was basically chosen because we had so many different mechanisms of actions here that were basically working on the cells. When we were therefore testing a broad range of ways of cardiotoxicity.
very successfully, actually. So I won't go into detail with all the compounds. I'll just show you a few of them, starting with pentamidine, for example. So here, what you can see here is the cell signal rising is when the cells are getting confluent on the plate and basically impeding current. But if you would apply a compound that would have a structural cardiotoxic effect, then you will see a decrease here. In case of pentamidine, you don't see that, which is actually a bit confusing because usually you would see that at least 24 hours after administration of the compound. But because the cell line has got no hERG, of course, you won't see any hERG trafficking block. And that's why it's actually a good control that pentamidine is not showing any results here. A proper control is allotinib that I would like to show you here, where we have strong ranging concentrations from 230 nanomolar up to strong 7.7 micromolar concentrations. And still you have no strong cardiotoxic, or actually no cardiotoxic effect, which you can also see here on the analyzed level. It's a nice cardio-safe compound showing no toxic effect on our cardiomyocytes. Now it's getting a bit more interesting. We have a cancer therapeutic agent here, vincristine, and we see concentration-dependent decrease once we added the compounds. So basically an increase in the toxicity that is also visible here when you look at the analyzed data. And that the higher the compound concentration is, the more toxic it gets for the cells. And the similar effect is visible very nicely on a concentration-dependent level for doxorubicin, of course, and anthracycline that is heavily cardiotoxic. And you can see here as well very nicely the concentration-dependent decrease in cell signal, basically meaning an increase in toxicity. And that was, to wrap it up, like I said, very nice to perform this study, especially because it was done in no time because of the throughput. And this, I would like to thank our partner Nanion Technologies just for great partnering in general, but also Fujifilm and Axel for awesome collaborations that we perform. And of course, last but not least, the FLEXcyte quiz. I hope you put some of your thoughts into the chat, because this is the result. We have isoprenaline on the left-hand side and the control condition coming after that, SBA on the right-hand side, and nifedipine on the far right. I'm sure most of you are right. Well done, you. And I'm open for questions.
Thank you very much, Bettina. Very nice talk. I was about to remind you, where are the quiz results? We already have some questions for you. So the first one came from Martin Trebert. Can you co-culture the smooth muscle cells with endothelial cells?
Well, this would definitely be something that would be a follow-up, because that was like an initial study. You know, we wanted to get everything in place, but yes, perfect question, and will be noted down, and we'll probably be able to present something like that in the next cardiac safety meeting.
I have one hand and one question written. Maybe I read first, and then Marcus, I let you sign in. So from Puya, I hope I pronounced that correctly. To evaluate the drug-induced changes in cardiomyocytes, the PASHCON technique can measure the cardiac action potential in all the individual ion channel currents. Then how would the measurement of cardiac contractility be important for preclinical drug toxicity screening?
I think it is always important, because what we do in preclinical drug development screening is we look at actually the three of them. We look at the electrophysiology, we look at the calcium transient, and we look at the contractility. And personally, I think the contractility gives you the best view, because the contractility is at the end of the whole contractions of this whole motion of cycles. So whatever you see in an electrophysiological level, you see as a contractile answer. What you see on calcium level, you see on the contractile answer. The other way around wouldn't work. If you have an effect on electrophysiology, but you, for example, would have that's fine. If you would have an effect on calcium, you wouldn't see that if you only do patch clamp. As of course you would see that too, but it's the best way basically to start it from the back, because then you can include all the others as well. So this gives me the that's why I think this is the most important way, to start basically from the last bit of the contraction cycle.
Or one does both.
Yeah,
that's also an option. Marcus, can you turn on your microphone and ask your question?
Hi, Birgit.
Bettina.
Hi, Bettina. Sorry, very nice talk. Sorry.
No worries.
I have a question. Can you estimate if the layer of cells, is that really a monolayer around in the FLEXcyte, or would you think it's more like a three-dimensional shaped orientation of the cells?
It is more monolayer because we always do an evaluation of the cell type, no matter which one it is, and then we choose basically the perfect conditions where we have the right amount of cells to get a confluent monolayer, but also not get too thick, and also just to basically save cells, because especially the commercial ones are, not can be a bit expensive, so you kind of have to find the right way in the middle. So what we present is monolayer.
Okay, thanks.
Thank you.
Thank you very much, Marcus. So more questions, really nice. Takasuna-san is asking if you can co-culture smooth muscle with endothelial cells. We expect that you will evaluate the acetylcholine-induced EDRF effect. Okay, well, that sounds interesting, but we haven't done that yet, so I can't answer that, but can't comment on that. But it's a good hint. Thank you very much. And the last one from Adam Hill, nice drug data on FLEXcyte. Are you aware of any data showing differences in contractility as measured on FLEXcyte for disease-causing variants versus normal cardiomyocytes? We do have projects going on for that, but at the moment I wouldn't share that right now, but maybe in the next, for the next safety meeting, I would. Thank you. Thank you very much, Bettina. So we were perfectly on time. Perfect. And very, very nice talk. Thank you so much. Thank you too. We will switch now to US. We will welcome Ravi Vaidyanathan. I hope I pronounced your last name. I always try and fail. Ravi will have a very nice continuation to the talk that Bettina just finished. Ravi is a senior product manager for cardiac portfolio in Fujifilm Cellular Dynamics. And what he will show is a little bit of twist to the talk that Bettina had, and he will talk about the effect of the fibroblast co-culture on the contractility of iPSC-derived cardiomyocytes. Thank you so much, Ravi, that you woke up so early to join us. Floor is yours. Thank you very much.
I hope everyone could hear me. Thank you, Elena, for that introduction, and you did say my last name correctly. Thank you for that. So today I'll briefly talk about what we are doing. It's more of an update from last time, what we have presented. To give you, oh, sorry, those two slides. Okay, so to give you a brief overview of the cardiovascular portfolio. What we have in our portfolio is iPSC-cardiomyocytes, which we have mainly in apparently healthy normal lines. We have 2 donors, and Adam, to answer some of your questions too, we have disease models in our portfolio. We have a DCM line that is available, also has an isogenic control that you can use for your studies. We also have engineered lines where we have engineered at least 2 different disease models. One is Brugada syndrome, and the second one is arrhythmogenic right ventricular cardiomyopathy. Then we have the cardiomyocyte squared, which is what most of the data that you saw from Bettina came from. We also have endothelial cells, and we have recently been working on iCell cardiac fibroblast, and this is what I'm going to mainly focus on going forward, and I'll give you a story on how that works. In addition to our cardiomyocytes, endothelial cells, and fibroblasts, we also have media. We have our serum-containing media that we have a plating medium and also a maintenance medium, and our cells really like these and have been used, and these media have been used for a lot of the studies that you are currently that were currently shown. We recently also launched the serum-free media, which is the iCell cardiotox assay medium. which is albumin-free, and iCell cardiotox serum-free medium, which is serum-free. So our cells are best known for working in 2D assays, and we are platform agnostic. We work with a variety of instrument providers. I've listed a few of them here. One of our major collaborators is Nanion. As you saw, innoVitro gave a really nice presentation on some of the work that they've done with our cells on the robustness and lot-to-lot variability. But in addition to that, we also have a protocol, an application note for a lot of the instruments. The one thing that I wanted to point out is that all of these instruments were used as part of the SIPA too at some point. And we were part of the original study. And if you remember, there were 10 sites that did a lot of work trying to validate the protocols, the cells, and we have been part of it right from the beginning. So there are also strategies to improve the functional maturity of iPSC-derived cardiomyocytes. And some of the things that most people want to do, and they like to do it, is to make cells look like adult cardiomyocytes. And some of the things they've been trying to do is to have a different culture medium, supplements, extracellular matrix surfaces, modify the surfaces such as ECMs, and also with patterning. Then there are devices that can do some of these improvements too. I'm going to mostly focus on the 3D and the co-culture aspect of it. So this is a study from AstraZeneca that came out in 2018, where they put cardiospheres together. And what they did is they combined our cardiomyocytes, iCell cardiomyocytes, with primary human cardiac fibroblast and primary human cardiac microvascular endothelial cells. And when they combined them and looked for specific markers of maturity, such as vimentin and cardiac troponin I, across day 14 and day 21, they saw that the cardiac troponin I was robustly expressing, and the vimentin was primarily expressed in the fibroblasts. They also saw matrix with collagen 1, which is shown on the left over here. In the study, they actually compared 15 drugs that were known to have structural toxicity, and 2 of the drugs are shown here, doxorubicin and sedative. And if you compare that to control, you can clearly see that the troponin I and vimentin levels are lower as compared to control. The one thing that they found from this really nice study was that the sensitivity was similar at about 73%, but the specificity was higher in the cardiac microtissue, which is shown in black over here, compared to monolayer of cardiomyocytes. The study mainly focused on structural, not on function. So they looked at ER integrity and mitochondrial membrane potential. And I think the gist of this was that there is some maturity that you see when you combine these 3 cell types together. So what we did, and this was presented at last time, is to combine 3 different cell types. We used our iCell cardiomyocytes, our iCell endothelial cells, and used a primary fibroblast. And when you combine them in the ratios mentioned here, and then feed them with a combination of our medium and the PromoCell endothelial supplement, we saw that there were quite a few benefits. But when we evaluated the market, customers were complaining that the cells, the fibroblasts mainly, were very variable from lot to lot. The expected results were not very consistent, and the cells were being very, very proliferative, and they would take over the culture over time. So what we decided was that it would be nice to have our own iPSC-derived fibroblast. So that's what we did. And here I'm showing you some data showing the purity and identity of our cardiac iCell cardiac fibroblast. We looked at the purity. They were very high pure when we measured them right out of thaw, and they were also alpha SMA negative, which means they are not activated. They express all the right markers. We are showing here collagen, preostin, and also connexin 43. These are all key cardiac fibroblast markers that our cells seem to express. So then what we did is we combined these 3 cell types into ours. So now you have an isogenic culture of I-cell cardiomyocyte squared, I-cell cardiac fibroblast, and I-cell endothelial cells, all from the same background. And when you combine them right out of thaw, they found these really nice spheres when you combine them in these 3 ratios, and we'll be providing the media for this. You have a week-in-free workflow. You do a 50% media change Monday, Wednesday, Friday. That's what is shown here. And in 2 weeks, they are ready for assay. These are some of the preliminary experiments that we did with the cardiospheres. On the left is an intact cardiosphere stained with looking for fibrotic necrotic core, and as you can see from the staining, there is the cells are really nice and viable from the trichome staining. The 10,000 total cell cardiospheres had really nice uniform shape, and they were between 300 and 400 microns. And on the right-hand side, what I'm showing you is a 384 well plate where on the edges we plated 5,000 total cells, combining 3 cell types that I just talked about, and in the middle, 10,000 total cells. And as you can see, they form really nice uniform spheres. We look within the same window. So these cardiospheres can be also amenable to high throughput assays. One of the key aspects that we were looking for is a positive ionotropic effect with our cells. So here, I'm showing what we've done on day 14 with the cardiospheres that we generated. Yellow is the control, red is under the drug condition. And as you can see in isopaternal and dobutamine, these are 2 positive ionotropes. You can see that compared to the yellow, the red, which also is faster, but also has a bigger calcium amplitude. And that is the same thing that we saw with dobutamine. And results from multiple experiments are shown on the right, where you can clearly see that that is at least a 1.5-fold increase in the amplitude of the calcium transient compared to control. This is some immunostaining that we had done with our collaborator molecular devices. Panel A and B, the red is cardiomyocytes, green in panel A is cardiac fibroblast. B is green is endothelial cells, and blue is nuclei. On the right, the colors have been switched. So red is cardiomyocytes, red is cardiac fibroblast, and green is cardiomyocytes. And I think what this comes out is that these 3 cells really seem to integrate really nice. They're nicely spread out, and they're forming these nice spheres. Now, how do you use this in any of the platforms that Nanion provides? So in 2020, AstraZeneca, again, published a paper where they assembled these cardiospheres, again, using our cardiomyocytes and endothelial cells. But what they did is they added these gold ion particles into the culture, and using a custom array of magnets, they were able to focus or bring those cardiac spheroids that were formed right onto the cardioexcite electrode, and their success rate was really high. So this is where you can see when this is cardiomyocytes only, cardiac myocytes, and cardiac fibroblasts with endothelial cells. When you combine them, they form these nice spheroids, and they've characterized some of that here. And this is showing that they're really nice and round with a red stain. They also are pointing in panel T to the gold ion particles, showing that they are nicely incorporated, and because of this custom array of magnets, they could focus them on the electrodes. And if you read the paper, you'll also see that they are very high success rate. One of the things that they did is the positive ionotropic experiment that I just talked about. They added 100 nanomolar isopaternol, and control is in black, and isobutanol increased the amplitude significantly, as you can see here. On the right-hand side, what they did is they normalized the amplitude, but one of the other things that also happens when you add such positive inotropes, it not only increases the amplitude, but it also decreases the duration of contraction because it's now beating faster. So with that, they were able to show that you can do this. Now, they used a lot of custom-made instruments. Since then, Cranier has now this magnetic tools for 3D cell culturing. Then they also have the nano shuttle, which has the beads that can be incorporated into this cardiospheres now. They have magnetic plates that comes in 624, 4896, and 384 well plates, so it's amenable to high throughput too. The other thing that you would probably need is to move these spheroids around. So they have these mag pens now that can be used to move one at a time, or you can move many of them at the same time. So this is how we think that now with these tools available, you should be able to use these cardiospheroids directly on the cardioexcite plates. And then repeat these experiments. I'm not going to go over the slide because Bettina did a really good job talking about the various aspects of the FLEXcyte system and the nice video that she had that I didn't have access to, but it's shown right here. But some of the work that we are doing right now is to see, as I said at the beginning of the talk, to see how these cardiomyocytes and cardiac fibroblast co-culture work. This is the workflow that we currently have, where you plate our cells on the FLEXcyte plate, maintain them for 1 to 10 days. You can dose them at 5 to 9 days, and then start recording for acute experiments within 20 minutes. I'm going to skip over this too, because Bettina did a really good job showing you guys how these compounds work, but basically the compounds have the right response on these plates. This is all the plates also measure true contractility. So that is where I think there is a lot of interest in these plates, at least from our end. We feel that the physiological stress that the cells see as compared to plastic or glass is more relevant. And that is where we are evaluating this in-house too. This is a very interesting experiment where Matthias and group were able to culture the cells for up to 44 days. And as you can see, some of the maturity markers, which is decrease in beat rate and increase in amplitude, were all seen in our cells when they were cultured for really long duration, even though the experiments can be started as early as day 6, day 7. So this is the data that I wanted to share with you guys today. One of the things that we realize is that it is not easy to take an existing protocol that I just talked about and just add something to see the results. This is something that we're realizing, and I'm sure most of you realize. So there is a lot of optimization that needs to be done. And we are at the beginning stages here, where we compared no cardiac fibroblast to we adding 10,000 cardiac fibroblast to 15,000 cardiac fibroblast to different concentrations of cardiomyocytes squared. And we are still working on the optimization, but some of the things that we are seeing early on is that when you combine these two cells, you start seeing an increased amplitude on day 4 and decreased beat rate. We would like to take these cultures longer, but we haven't had enough time to look into details of this. And I'm hoping that if I'm given this opportunity to present again next year, you will see more details and probably an application protocol on how to use these cardiac fibroblasts in 2D with our cells to see some of the benefits of co-culture and 3D modeling that might be relevant. So basically what I've shown you today is that combining iCell cardiomyocytes, iCell endothelial cells, and iCell cardiac fibroblasts directly from thaw can generate 3D triculture cardiac microtissue efficiently. These microtissues demonstrate spontaneous beating and have a robust calcium signal. These microtissues also show positive inotropic effects, suggesting that iCell cardiomyocytes are further maturing in the 3D environment. The other thing was that the cell cardiac spheres can be used on the cardioexcite system using the magnetic options that are available right now. And then finally, we are working on optimizing the co-culture of iPSC-cardiomyocytes and iPSC-cardiac fibroblasts on the FLEXcyte, and we're hoping that we'll have something available soon. So thank you, and I can take any questions.
Thank you very much, Ravi. It's a lot of new things that you have there in development. Thank you for showing them to us. Do we have any questions? Okay, from Stephen, the first one. Hi, Ravi, nice talk. For the cardiospheres, do you have a sense of the relative transducibility of these spheres in potential to evaluating the downstream effects of AAV gene therapies?
So we are actually looking into that right now. We have already collected some data that actually I presented at ASGCT on 2D. We haven't done it in 3D yet, but we are looking into it, and we expect that it should be possible because you're using them right out of thaw in suspension. You should be able to infect them right at the beginning when you thaw them in suspension that most people do. So yes, it's possible. That's the best answer I have. I don't have any data, but I'm happy to share the 2D data that we have with our cardiomyocytes and all the AAVs. We've looked at all the way from 1 to 10 with our cells.
Mm-hmm. I have one from one of the private channels. The cells that you have measured, they are atrial or ventricular cardiomyocytes, or do you see any difference when you mix them with fibroblasts?
So iPSC-cardiomyocytes is a mixture of 3 cell types for the most part. It has majority of them is ventricular origin. I would say anywhere from 50 to 70% is ventricular. We do have some about 20% atrial and then a few nodal cells. That is what gives them the automaticity that most people are dependent on to do their assays right now. So as of now, we don't have an answer on if there is any differences. But what our model brings to the plate is a whole heart, basically, you have in a dish rather than specific ventricular or atrial cardiomyocytes. But this is where it gets challenging when you have so ventricular cardiomyocytes, as you know, they don't spontaneously beat, at least the mature and good ones. We have a mixture of cells so that we can support the current field with automaticity. So that's where we are at. So I hope that answered the question.
Thank you. And from Shan, I guess, how big is the microtissue? Can it be frozen and thawed for use rather than to co-culture each time? So could you freeze them and then use them later, thaw them and use them later?
So we haven't tried it, but we haven't tried it yet, so I can't really. When you say freeze it, I'm assuming liquid nitrogen, and because they're a mixture of 3 cell types, I don't know how they would behave because of the connections that they make. It would be very tricky, but something that. So maybe I should answer this way. Currently, there is technology available that you can cryo embryos, right? I'm sure if that is available to us, then yes, you should be able to do it, but we'll have to be evaluated. Short answer.
Thanks, Ravi. And again, from Takasuna-san, did you confirm that co-cultured cardiomyocytes are matured not only functionally, but also gene expression or morphologically? That are related to maturation. So did you use any other techniques?
We did actually. So I didn't put a lot of data here because I was not focusing on the cardiac microtissue, but we have done some EM and we are seeing some caveolae, which is very exciting. You should see some of that data soon when we launch in a couple of months here. So that was one. The second is we have also looked at the distance between the sarcomeres. Most iPSC cells have them in the range of 1.2 to 1.5, which is immature, of course. We are seeing in the range of 1.7 to 1.8. So we are seeing some structural maturity. We are also looking into other aspects, metabolic maturity too. All of that is being evaluated right now. So I hope that helps.
Thank you. I have, there are a couple of minutes left and I have two questions.
Sure.
So you, at the last slide, you showed these strong or the higher contraction force traces after the fibroblasts are added to the culture, if I understood correctly. Do you assume that fibroblasts would then induce higher maturation because you add them there, or you don't know that yet?
So we don't know that yet. I think one of the things that we would like to do is to take these cultures long term, right? We're looking at 4 days mainly because we, when we try to do this we haven't tried very hard on getting these long term yet. We've had the system working for only few weeks, as Elena, you are aware. So we haven't done this. It's too early to say what are the fibroblasts doing. Some of the things that we're trying to do is, the 3D is easy, but you also need specialized equipment to do it. And we're trying to make it easy for our customers by if they can keep this current workflow and add more things to it, like co-culture, and see if it benefits. So that was one. So that's first part. Second is maturation. Cardiac fibroblast, because they release the factors and also set down the matrix, we think that all of that is really helping the cells feel at home and in a more physiologically relevant environment, which already the FLEXcyte is providing, just better. So I think that's where we are seeing some of these benefits, but it's too early to say if the fibroblasts are only sufficient. We are also trying to see if we want to put endothelial cells in there into because our endothelial cells do form tubes. So it'll be interesting to see how all of that, falls into place.
And very shortly, because you always have these really nice colorful pictures, and you had really nice ones from the spheroids that you've shown. How homogeneous are they? So when you produce them, do they show similar homogeneous levels?
Yes, they do. So one of the things that we do is you combine all the 3 cell types in the right ratios, as I had shown into a dish. So it is one tube that has all the 3 cell types, and then you just pipe it from there into your well. So they're very homogeneous, and they continue to stay homogeneous over the 14-day period. And this is what some of the complaints that we've got with the primary fibroblast, because of the lot-to-lot variability, and also variability in the donor, because you don't get the same donor. We are seeing that with the iPSC-derived fibroblast, you are seeing better control, better homogeneity, And then the second thing that we are also, you can use this model for is to look at fibrosis. With primary cells, because you have no control. They say it is apparently healthy normal, but there are no healthy volunteers for fibroblasts. So, there is always that needs to be taken into account when you're using these primary cells. And I think that's the void that these iPSC-derived fibroblasts are going to fill.
Thank you very much, Ravi. Thank you for joining us and for your nice talk. And now we continue. The last but not least, Khuram Chaudhary is joining us also from the US. I don't think I need to introduce you a lot, Khuram, everybody knows you very well. What I can say is that after many years at GSK, you are now Scientific Senior Director at BMS. So congratulations for your new adventure. Thank you very much for joining us. And today, Khuram will have a very nice overview about the results that have been obtained on this various platforms at HESI Stem Cell Working Group. talking about chronic drug-induced cardiotoxicity assessments of using human iPSC-derived cardiomyocytes, and what is really interesting, also human heart slices. So I'm very excited for your talk. Thank you so much for joining. The floor is yours.
Can you hear me okay?
Yes.
And is my screen visible to everybody?
To me, yes. So I guess to everybody else as well.
Okay, very good. Thank you again for the invitation. Let me just cue everything up. Okay, so I want to thank Nanion and my friend Sonja for this invitation to speak at the symposium. There's some very interesting talks we've heard already today. I'll be providing an update on a global collaboration that including many of you who are on the call today under the hERG umbrella, which is the Health Environmental Science Institute. Our goal is to assess chronic treatment-induced cardiotoxicity in preclinical development. And that's something that I think many people have worked on, but in this collaboration, we're really coming up with a better way to do the preclinical development. So let's see, okay. All right. So to begin, I'll share a little background on drug development and the causes of failure that we see in non-clinical and clinical development, the mechanisms associated with long-term toxicity that go beyond ion channels alone, which is the standard ICH-S7 supportive study. And we'll follow that up with a very high-level update on the stem cell working group collaboration. So as part of the background, we all know that drug discovery and development is an exercise in failure. We have very few lead molecules early in discovery that actually make it to the clinic. But what we know is that the cost of failure becomes exceedingly high if that failure occurs later in development. And so it's sort of a paradox there. The major causes of attrition haven't changed for decades, and they include more than half of that attrition due to efficacy and human and animal safety and toxicity. And over the past decade, regulators and industry have emphasized the investigation of ion channel dependent QT prolongation. As you know, many of you have talked about SIPA today, as well as what you've heard recently over the last year in terms of the ICH-S7 E14 Q&As and the best practices that were adopted. However, adverse drug reactions often occur or are reported for cardiovascular events, including heart failure, myocardial disorders, changes in blood pressure, structural changes, including fibrosis, as well as vascular injury. And so it's not just a matter of understanding the acute cardiotoxicity, rather what might happen over chronic treatment with respect to function and structure. Designing the preclinical strategies to assess that risk, though, can be very challenging, as most models require the use of animals dosed chronically over time, and then sort of a retrospective analysis of what mechanisms were modulated. So here I've cited a recent review article that identifies various mechanisms for drug-induced cardiotoxicity. And what you can see is that that goes beyond just ion channel pharmacology. These include signaling pathways, growth factors, energetics and mitochondrial function, apoptosis, ion channel trafficking, and changes in mechanical function, including fibrosis, as well as we mentioned earlier, electrophysiological changes. As you can see, there's multiple drugs that have been withdrawn from the market due to cardiovascular toxicity, and that goes beyond just the ion channel and QT prolongation. And these drugs actually span therapeutic targets and organ systems. So it's not just the cardiovascular specific therapeutic area or indication that we're concerned about, but it's drugs that are, for instance, anti-neoplastics, anti-cancer agents, GI as a target, anti-inflammatories. So it goes well beyond just understanding what happens in the setting of cardiovascular disease as a target indication. So the advantage of working in a partnership like HESI is that they're able to bring together industry scientists, regulators, academicians, and CRO partners to address a specific concern. So the goal of the stem cell working group was to identify relevant endpoints that could translate to clinical cardiotoxicity. We also had the goal of selecting an appropriate reference set of compounds with specificity to benchmark these endpoints. And you can see those here. There's about a dozen here. And you can also see the endpoints that we were interested in capturing. we wanted to test the hypothesis that might bring up a signature or a fingerprint of chronic treatment-induced cardiotoxicity, including both structural and functional tox, using non-clinical in vitro assays. And so what we've done is engaged with greater than 20 different partners across industry, academia, regulatory, and CRO contributors. And the study design for this was actually quite prescriptive. So the goal was use similar reagents, capture the data at similar time points, whether that be 1 hour post-treatment to understand the acute cardiotoxicity versus 24, 48, 72, and so on. But also then to look at different endpoints across these platforms. And so what I'm going to do now is share with you some of those technologies that we used, especially some of the more innovative ones. So here we have a subset of technologies and reagents employed in this collaboration. This helps illustrate the breadth of the effort. First and foremost, the goal was obviously to use in vitro and ex vivo systems, including stem cell-derived cardiomyocyte monolayers. You can see that here, engineered heart tissues, as well as using primary human myocardial slices. And so when you look at some of the endpoints captured here, you're probably relatively familiar with them. And those include microelectrode arrays, contractility measures for monolayers, changes in action potential using fluorescent indicators, actually measuring physical force and contraction from the tissue preparations. And so again, the durations and the sampling intervals were very prescriptive, but each individual contributor was able to use their systems however they saw fit, obviously to manufacturer specifications or to some level of maturity that we could say was steady state, and that was representative of cardiac physiology. So I'm going to further illustrate some of those technologies by showing you examples of things that are interesting and kind of span the chronology of drug development from that early hazard identification through to looking at signaling cascades and mechanisms. And so here, this contributor suggests a representative trace from a bright field imaging platform. We have an instrument that captures that bright field image from a dish of a monolayer of cells. It uses landmarks associated with bright field imaging from those cells. And as that landmark or the speckles change, that deformation is picked up as a waveform. So that waveform then allows us the measurement of relevant endpoints such as beat rate, the contraction velocity and relaxation velocity, amplitude and relaxation parameters. And as you can see, these measures change with chronic exposure. And in this case, we're looking at doxorubicin, which is one of our positive controls, as well as in a concentration dependent manner. And so we hope that the results here mimic what we see in terms of progressive cardiotoxicity and a decrease in function that's observed in patients who are taking doxorubicin or other anthracyclines, which we know cause a progressive reduction in cardiac contractility. So this slide is a little bit more animated, so I'll run through all these different steps. In this assay, it's a plate-based assay with higher throughput. We use stem cell cardiomyocytes that are cultured with maturation stimuli. So in this case, the extracellular matrix has various factors that enhance maturation. You can see on the top right, you get a very striated sarcomere pattern. You also get binucleation of the myocytes, which again suggests some level of maturation. And so using this high throughput system, you can actually, can't run the, oh, for some reason I can't run the video. But what you'd see here in the 96 well plate is that the entire plate flashes, and those flashes are a measure of the calcium transient as picked up due to a transduced protein called GCAMP. This is a calcium indicator protein. And so in real time, we're able to measure that calcium signal, which is a surrogate of contractility. And you can see that as you capture that over time with your concentration dependence, you see some progressive decrease in the calcium homeostasis. So again, this is a measure of that progressive drug-induced chronic treatment induced cardiotoxicity. So we also employed many of the technologies you're familiar with in assessing acute cardiotoxicity. And that includes the microelectrode array, an impedance measure for contractility, as well as novel assays such as the FLEXcyte, which culture cells on a flexible membrane and actually allow us to measure contraction. So again, I'm very cognizant that we highlighted the sampling intervals, because again, we want to make sure that we're capturing our data in a sort of a harmonized way, even though the assays are very protocol specific and unique for their endpoints. And so here's some of the representative traces. On the left, you can see there's field potential shortening due to the exposure of this compound. And that sort of falls in line with what you would expect in terms of contractility. Oftentimes, calcium channel blockers will shorten the field potential duration or action potential duration, and you'll see the decrease in amplitude in your impedance measure. And you can also see that over time in your FLEXcyte. And so these assays are confirmatory of each other. And so now not only are you looking at one endpoint, but by measuring several endpoints in parallel, you can actually come up with a signature, or sort of, like that chicken or the egg phenomenon, like which mechanism is actually leading to the depressed function. So as we move on, we're looking at more complex outputs, right? So in this case, the contributor was measuring field potential duration as well as that functional change, but then using the Luminex to actually measure biomarker release. So we have an assessment of biomarkers such as troponin, creatine kinase, BNP, over the different treatments, and that allows us to assess cellular damage. So if you look here, you can see some of those mechanisms of changes in contractility that you'd see over time. So we have creatine kinase release, FABP3, BNP, NT-proBNP, and that's measured over all of the different treatments over time. And so in this case, we saw that biomarkers such as BNP were released in troponin, and that's exactly what you would see in a clinical setting, right? So the first indication that you have some type of cardiac dysfunction would be a measurement of troponin as a biomarker in your bloodstream. And then you'd go on and do electrocardiogram as well, and then sort of get to the root of whether you had an infarct or whether there was some progressive disease happening in the heart. And so this is the type of thing that we don't often see until late stage in vivo studies. So finally, we hope to investigate the mechanisms by which chronic toxicity occurs, including metabolics, energetics, mitochondrial function, and contributors here were able to measure metabolite production, such as lactate and arachidonic acid. We also looked at microRNAs, which are sort of a newer biomarker for cardiac disease, knowing that these, when measured by PCR, are indicative of cardiac damage. And this is just a sample of that data. Again, I'm not going to go into too much depth on this because the results are just now coming in. We have a statistician who's working to collate all of that, and then we're working very closely to design that fingerprint, as you might call it, for that preclinical development signature. And so if you look at this data, what you have is concentration dependence in the various biomarkers and metabolites, also the microRNAs, and again, we're looking at that over time. So the next thing I want to show you is how you take that functional endpoint as well as the biomarker and come up with some sort of signaling pathway or mechanism that explains why you have that progressive change in function. So in this example, the contributors are using the seahorse assay to measure mitochondrial respiratory capacity as well as stress, and that includes oxygen consumption in parallel to the functional endpoints. So analyzing the mitochondrial oxygen consumption in parallel to those functional changes gives you that mechanistic approach and a signature to the compound-induced toxicities. So you can imagine that when you take this and you develop some sort of heat map or some sort of principal component analysis, you can actually come up with not only the progressive change in function over time in vitro, but a potential mechanism by which that occurs. And so additionally, the measurement of mitochondrial membrane potential provides further insight into the compound-dependent effects that may create things like reactive oxygen species, things that may inhibit the electron transport chain, leading to progressive changes in function. And so we hope to create a signature which you can differentiate that chicken or the egg with respect to the cause of the cardiotoxicity. And that may otherwise only be investigated retrospectively after you've seen something in a late stage development study in vivo. So in summary, while significant emphasis has been placed on the non-clinical assessment of acute cardiotoxicity, we've all heard about the ICH S7 Q&As and the E14 best practices. Less is known about the chronic treatment-induced effects, including structural cardiotoxicity, prior to late-stage in vivo studies. So the goal of this project is to establish that predictivity, sensitivity, and specificity of various platforms that use novel reagents like stem cell cardiomyocytes, as well as primary human cardiac tissue as a benchmark, ultimately to understand what happens in terms of chronic actions of drugs. And so before I conclude, I want to make sure that I thank all of the contributors. You can see there's a wealth of knowledge here. There's a multitude of contributors across industry, academia, regulatory, and CROs. On the right alone, you see we have over 20, upwards of 24 different contributors working on this. It's been a global project, again, supported by HESI. And I think we're at the point now where the results have come in. We're at the point of doing statistical analysis. There are novel ways to visualize that data, and you should be hearing something at various conferences coming up. We started with SPS last year. We tried to do a presentation at SOT. There's some work coming around for Safety Farm Society later this year. And you should see some output from this as part of the stem cell working group. So I want to thank you for your time. I appreciate the invitation, and I'd like to open it up to any questions.
Thank you very much, Khuram. This was wonderful. I enjoyed a lot. Do we have any questions? Anyone writing anything in the chat? If not, I can ask two. We have one from Sonja, actually. So she's asking how far did the analysis of the data go? Can we expect a publication soon, maybe?
Yeah, that's a good question. We have a sub-team that's dedicated strictly to the data analysis. You can imagine that while each platform is different, we were very prescriptive on how the data should be submitted. So we had a group come up with a table format where we divided up various endpoints, the time points, each assay parameter. And so just on sort of field potential alone, there's 3 or 4 different endpoints. And you can imagine across the various platforms, there's a multitude. And they're all different. So we're trying to harmonize that. But we've got a really good team of statisticians and scientists working on this. I've seen some of that analysis already in heat map format. They're writing some scripts on how we can automate that analysis. There's a sub-team of individuals who volunteered to do that analysis as well. So I wouldn't be surprised if you see something within the next year in terms of a publication. Well,
that's good. Very fast track, I would say. And I have one for you, if you don't mind. This is a very comprehensive study, enormous amount of work, a huge team. What kind of insights did you personally get from all of this data that you have performed? What would you think would be the most interesting that you have seen so far?
Yeah, I think we took a very agnostic approach or an unbiased approach to coming up with the assays, reagents, platforms. So no bias going in to say, well, like stem cell cardiomyocytes are immature, or we only need dog or monkey tissue, or we only need human tissue. And so when you take that approach, what you see is there will be endpoints that are not sensitive. There will be reagents that are not sensitive, and you'll find those that are. And so the goal here is not to be that prescriptive. It's one, to assess the field and say, in vitro technologies and novel approach methods have some utility in assessing chronic cardiotoxicity. This has a potential impact on 3 Rs, so reduction of animal use, being more predictive of the clinical setting. So as long as you're open to all that, you'll find some assays will be more sensitive, some will be less, and it'll really help us to design a more appropriate, clinically relevant strategy early in drug development.
Thank you. And now from Ron one, it starts a little bit funny. Without having a crystal ball, do you think the regulatory authorities will incorporate the newer assays, and when?
So I don't I mean, I think it's a really important distinction. Much of the work that we're doing in HESI is meant to be exploratory, investigative, and to help people better understand how to make safer molecules for patients, right? Unmet need. There's obviously a regulatory component because they are our partners. We want to ensure that when we submit this data, it's done in a manner that's relevant and they can interpret it. I think there's quite a bit of buy-in from regulators. In fact, if you look at the concluding slide, there's several individuals from the FDA who we've partnered with who are generating some of that data as well. I think everybody's familiar with the FDA Modernization Act. So again, there's a culture right now of shifting to novel approach methodologies, organs on chips, microphysiological systems. And I think that's the future. So I think there's quite a bit of regulatory buy-in.
Okay, from Katerina, one more. Hi, great presentation. She has two questions. What is the first time point you consider as chronic one? And have you already published some protocols for the chronic cardiotoxicity screening if someone would like to follow it in their studies?
So let's start with time points. What you'll probably intuitively understand is that this is not an in vivo setting. And so we don't have the same drug disposition properties. We don't have the same metabolism, the clearance. And so, a PK profile in an animal would look like, the sine wave. Here, we're just adding compound every 2 days. We add fresh compound and we change the media. So it's not truly a mimic of an in vivo study. It's really a matter of having that chronic exposure and eliciting signaling pathways that could be detrimental over time. So it's hard to say what is a 1-for-1. Is a 7-day rat tox study the same as a 24-hour, in vitro treatment? I don't know. Again, this is hazard identification. So that's a tough question, but what we've chosen to do is work within the boundaries of the reagents as well. So these stem cells, for instance, have to be fed every 2 days, or we like to think that they're fed every 2 days. We're also using serum-free and serum-containing conditions. That has an effect on the physiology. But every 2 days, we replenish the compounds, and we've gone out to 96 or the next 24-hour increment, so up to 4 or 5 days in terms of looking at the effects. Again, it's really a matter of seeing that progressive change, and that's the hazard ID. So that's a generic way of answering your question. And so the second part was how much of it's published. Well, keep in mind, we're trying our best to stay within the protocols or the methodologies that have been given to us by the reagent manufacturers and the assay platforms. And it's a matter of doing things in that controlled prescriptive manner, but looking over time. We're trying not to change any of that culture condition just to see what the sensitivity would be. Let's just see what the output's going to look like. I don't think we've done this in a concerted way like this effort ever. So that's yet to be determined.
Thank you, Khuram. And one more from Stephen. Is it reasonable to assume human iPSC-cardiomyocyte would model human cardiotoxicity in response to drug versus isolated mouse cardiomyocytes?
I think that we're going to have a conversation over dinner or a coffee. It's a very long answer. I think there is utility in using primary mouse cardiomyocytes, but I think we all agree you can go back to the early 1900s and find that the physiology of the mouse and the human are very different. The calcium homeostasis is very different. The acute function is very different. EC coupling is very different. EC coupling gain is very different. So I can go on and on and on, but it's a living organism. It responds to drug treatment, and it's often a preclinical species, non-rodent or preclinical rodent species used in drug discovery and development. So there is utility. I think what we're learning is that if you go just to that pie chart I showed you, one of our biggest problems with drug development is efficacy. Is it because we're only testing on mice? Possibly. And so the human relevance, I think, is very important. And what we're trying to do is establish human relevant assays that have human proteins that reduce the use of animals and that give us better clinical translation. So there's a place for all of this.
Very nice way to conclude the meeting, I think. And thank you, Khuram, for your talk and for the discussion. I will now, for the closure of the meeting, just share a couple of slides from my side before we say goodbye for this year. So I will try to share my screen now. Let's see if that will be possible. So I hope you see my screen in full view. Yes, so just to maybe raise a little bit of attention of some news or changes in our image that you might have noticed lately. So we have a new website that we've launched 2 months ago. And I would like to invite you to visit it and maybe look at these 2 areas here. So the application areas and therapeutic areas. We have tried to finally curate information, not only from us, but also from other scientists for various, for example, disease models, for various application technologies, and so on. And one of them is cardiac safety. So we have a whole page dedicated for cardiac safety, which I would invite you to visit. And before we all leave this meeting, I would also give a little bit of shout out to other meetings that are coming. One of them is not organized by us, but is approaching rather fast. The deadline's actually, I think, on 10th of June already for the abstract. If you're all coming to Safety Pharmacology meeting in Brussels this fall, please join us. We will have posters, we will have our booth there as well. But also in October, we are hosting our Nanion user meeting here in Munich from 24th to 25th of October. You're all kindly invited to join us. And at the end, I would like to thank you, every one of you who joined, especially our speakers. These talks were wonderful. They covered a lot of topics. And I think our speakers joined us as well as you attendees from, I think, every possible time zone. So thank you very much for staying late, waking up early. These were wonderful talks. Have a wonderful day or evening, wherever you are. And hopefully we see each other in 2 years for Cardiac Symposium 2025. Thank you very much. And we will follow up with recording of this video, so everyone who registered will get a copy. Thank you very much, and bye-bye.